Have you ever really paused to consider the intricate, almost hidden world flowing within your very veins?
Hmm.
I mean, beyond the red blood cells or platelets we all know about, there's this incredible, silent symphony of proteins, just tirelessly working, keeping everything running smoothly.
Absolutely. It's a universe in itself.
Exactly. So today, we're embarking on a real deep dive into this fascinating, complex universe of plasma proteins and
And
immunoglobulins.
this isn't just, abstract biology.
No, not at all. It's fundamental. It's about understanding the very foundation of your health, how disease works, right down to how your body fights off infection or just maintains its vital balances day in, day out.
It's core physiology.
So, our mission for you today, well, it's pretty ambitious. We want to equip you with the kind of knowledge, the depth of understanding that an elite professional in this field possesses. We're going to pull back the curtain on these silent workhorses swimming in your blood. We'll meticulously explore their diverse roles, their really intricate regulation, and crucially what happens when that delicate balance goes, well, sideways.
When things go awry.
Precisely. So by the end of this deep dive, you'll gain a comprehensive, really actionable understanding of why these proteins are so vital, how they work together, and how your body defends itself at a truly fundamental level.
And to really achieve that mastery, we've grounded ourselves in a key source, Harper's Illustrated Biochemistry, specifically Chapter 52. It's packed with the kind of precise detail we need.
Okay.
We're going to cover, well, essentially everything from the basic multifaceted functions of blood itself,
Right.
all the way to the intricate, almost choreographed dance of iron homeostasis, and then, of course, the remarkable machinery of the immune system.
That immune piece is huge.
It is. And we'll examine the fundamental roles of key players, like, say, albumin and transferrin. We'll delve into the astonishing specifics of how your body actually generates antibodies.
The diversity problem.
Exactly. And unravel the protective cascade of the complement system. This whole deep dive is really crafted to ensure you don't just understand, but genuinely master every concept we touch on.
Yeah, get ready for some real aha moments, I think. Did you even know that the proteins flowing through your blood aren't just like passive delivery trucks?
Far from it.
They are critical architects of your immune defense, meticulous shapers of your body's fluid balance, even precise regulators of your metabolism. It's so much more intricate and, well, intelligent than just blood carry stuff.
It truly is. It speaks to this incredible silent intelligence working within our own bodies constantly, #bad1, the foundational roles of blood and its proteins.
Okay, so let's begin right at the foundation. Let's
Yep.
uncover the foundational roles of blood and its amazing proteins, because they truly are the unsung heroes swimming around in your plasma.
You really are.
When we talk about their biomedical importance, we're not just describing, random bits floating around. These are highly specialized molecular machines. Each one has a crucial predefined mission for our physiology.
Absolutely. Highly specific functions.
Take albumin, for instance. It's the ultimate multitasker, isn't it? Like a universal shuttle service within your bloodstream.
A workhorse protein.
Totally.
Yeah.
It facilitates the transport of fatty acids, steroid hormones, and just countless other vital ligands, making sure they get to the right destinations throughout the body.
Critical delivery.
Then you got transferrin.
Yeah.
That acts like iron's own personal courier service, meticulously aiding its uptake and precise distribution exactly where it's needed.
Very tightly controlled.
And of course, fibrinogen. That central building block for blood clots, a truly readily mobilized component, just poised to spring into action whenever your body needs to stop bleeding.
Right. And building on that, the whole process of blood clotting, it's not just about plugging a hole, as many might think.
No, it's way more complex.
What's truly fascinating is that it's triggered by this highly sophisticated cascade, a cascade of latent proteases. These are enzymes that start off inactive.
Vanagines, right.
Exactly. Zymogens, or blood coagulation factors. This cascade is just a beautiful example of biological precision. One inactive enzyme activates the next, which activates the next in this controlled, stepwise fashion. A chain reaction. And this ensures that a clot forms only exactly when and where it's desperately needed. It prevents widespread, potentially really harmful clotting throughout the whole circulatory system.
Like a failsafe.
It's a marvel of controlled activation, truly.
And then protecting us from the very tools that are meant to heal us. We have the body's internal bouncers, you could call them, protectors.
The inhibitor.
Proteins like antithrombin, A1 antiproteinase, and adenocroglobulin. Their crucial role is to shield our healthy tissues from, well, rogue proteases. These powerful enzymes are, paradoxically, essential for cleaning up dead cells or fighting invaders. But, they could cause severe damage if they were just left unchecked and unregulated.
They need tight control.
Absolutely. Antithrombin, in particular, plays a critical role in making sure blood clots stay precisely confined to the immediate area of a wound, preventing them from dangerously spreading, uncontrollably through your entire circulatory system.
Containing the response.
These proteins are truly vital guardians of our tissue integrity.
Which leads us to a really important question. How does the body so intelligently differentiate between friend and foe? How does it identify dangerous invaders while leaving healthy cells completely untouched?
Yeah, the self versus non-self problem.
Exactly. Circulating immunoglobulins, which we also call antibodies, form the sophisticated front line of our adaptive immune system.
Is specific defenders.
Precisely. They act as highly specific recognition molecules that can target and neutralize threats with just incredible accuracy. These molecular sentinels are constantly patrolling your bloodstream, ready to flag anything that doesn't belong, initiating a tailored defensive response.
Your blood, quite frankly, it's nothing short of miraculous when you think about it.
Yeah.
Its functions extend far, far beyond just simple transport.
Oh, absolutely.
You really could think of your blood as the ultimate, meticulously managed smart city infrastructure.
That's a great analogy.
It's not just the roads for transport, right? It's also the highly efficient waste management system, the precise climate control, the robust defense network, and the critical communication lines, all seamlessly integrated into this one flowing medium.
All happening simultaneously.
Yeah, like respiration. It's delivering life-giving oxygen from your lungs to every single cell and tissue, while at the same time carrying carbon dioxide back to your lungs for expulsion.
Essential gas exchange.
Nutrition. It's the delivery system for all the absorbed food materials, ensuring vital nutrients reach exactly where they're needed for energy, for growth,
Fueling the body.
then excretion.
Yeah.
Blood diligently transports metabolic waste products, the cellular garbage, to your kidneys, lungs, skin, intestines for removal.
Waste disposal. Critical.
And it's absolutely crucial for maintaining your acid-base balance, keeping your body's pH within that really narrow, healthy range.
Tight regulation there, too.
Water balance is another key function, meticulously regulating the fluid exchange between your circulating blood and your tissues, preventing, uncomfortable swelling or dangerous dehydration.
Fluid dynamics.
And let's not forget temperature regulation. Blood helps distribute body heat throughout your system, keeping you at a stable internal temperature.
Thermoregulation, yeah.
And of course, it's a primary defense against infection thanks to those vigilant white blood cells and the circulating antibodies we just touched on.
The immune surveillance role.
Plus, it transports hormones, regulating metabolism, ensuring your body's complex chemical reactions are perfectly coordinated. It carries various metabolites, the intermediate and end products of metabolism, and as we discussed, it's absolutely essential for coagulation, that intricate process of blood clotting.
It really is a multi-talented living fluid.
Totally.
But what's particularly striking, I think, is how quickly even seemingly minor perturbations in these intricate systems can cascade into really serious health issues.
Right. The consequences can be severe.
Consider deficiencies in clotting factors, for example. These lead to excessive bruising and bleeding, famously seen in conditions like hemophilia, where even minor injuries can become life-threatening because the body just can't form stable clots.
Devastating for those affected.
Or take Wilson disease. Here, a lack of functional ceruloplasmin, the body's main copper carrier, causes copper to accumulate to toxic levels in tissues. This leads to severe hepatolenticular degeneration affecting both the liver and the brain.
Copper overload, basically.
Precisely. Emphysema, that debilitating lung disease, is frequently associated with a genetic deficiency in ALA1 antiproteinase. This leaves the delicate lung tissue vulnerable to relentless proteolytic damage from enzymes like neutrophil elastase.
Lungs basically dissolving. Terrible.
And then there's this incredibly broad and complex category of autoimmune disorders. It's estimated more than 1 in 30 North Americans suffer from conditions like type 1 diabetes, asthma, or rheumatoid arthritis. These are characterized by aberrant immunoglobulin production. The immune system mistakenly attacks the body's own healthy tissues.
The immune system turning on itself.
Exactly. And furthermore, immunocompromised individuals, those with insufficient protective antibodies, perhaps due to conditions like HIV infection infection or the administration of immunosuppressant drugs are rendered profoundly vulnerable to opportunistic infections. It really illustrates the critical role of these protective proteins.
So what does this all really boil down to for us? While some diseases, like, say, hemophilia, are relatively straightforward in their cause, a missing factor.
A single gene defect, often.
Right. But many autoimmune disorders, they're a cryptic interplay of complex genetic factors, dietary influences, nutritional status, environmental triggers, even medical history. It's a true deep dive problem for researchers trying to unravel why the immune system tragically turns on itself.
It's multifactorial and incredibly complex.
That really highlights the incredible, often surprising interconnectedness of our bodily systems and how a seemingly small malfunction in one area can just ripple outwards with profound and often devastating consequences. It truly underscores the body's delicate balance.
Beautifully put. #tag2. The intricacies of plasma protein composition and dynamics.
Okay, now let's peel back another layer. Let's delve into the fascinating intricacies of plasma protein composition and dynamics. Plasma is truly a complex mixture of proteins, far more so than early scientists could possibly have imagined.
Definitely. Historically, using relatively rudimentary techniques like salting out with ammonium sulfate.
Kind of a brute force method.
A bit, yes. Early researchers initially classified plasma proteins into just 3 broad groups: fibrinogen, albumin, and the globulins. This was a foundational first step, of course, providing that initial glimpse into this complex fluid, but it was really just the beginning.
Right, scratching the surface.
Exactly. To build on that, while those early methods were groundbreaking for their time, clinical scientists dramatically refined our understanding with electrophoretic separation.
Using electric current to separate them.
Precisely. Using techniques like cellulose acetate electrophoresis, they discovered they could separate serum proteins into 5 major distinct components: albumin, then the AO1, albumin, and globulins.
You can actually see the bands on the gel.
You can, and this ability to visualize these distinct bands was a huge leap forward for medical diagnosis. It allowed clinicians to observe patterns and changes indicative of various diseases.
The diagnostic fingerprint, almost.
In a way, yes. Furthermore, when we examine their molecular structure, we find that plasma proteins are often rich in disulfide bonds. These are absolutely crucial for maintaining their precise 3-dimensional shape, their conformation, and their stability.
Holding them together.
Yes. Many are also frequently glycosylated, meaning they have complex carbohydrate chains attached, forming glycoproteins. Others are lipid-bound, creating lipoproteins, which are vital for lipid transport throughout the body.
Sugars and fats attached,
Yeah.
adding complexity.
Just look at the incredible range in their size. Hemoglobin, for instance, comes in at around 64.5 kilodaltons. Albumin is about 69 kilodaltons. But then you jump to fibrinogen at a much larger 340 kilodaltons.
Wow.
And then 1,1 lipoprotein, a massive complex, weighing in at a staggering 1.3 megadaltons.
Huge difference.
This immense variation in size and composition directly reflects their incredibly diverse and specialized roles within your body.
And these proteins, their sheer presence is absolutely essential for fluid distribution and what we call oncotic pressure, right? Sometimes called Starling forces.
Correct. This is a core concept for understanding how fluid moves around your body. The aggregate concentration of proteins in your plasma is typically around 7 to 7.5 grams per deciliter.
That sounds like a lot.
It is. And this concentration creates an osmotic pressure, or oncotic pressure, of approximately 25 millimeters of mercury.
Okay, so what does this pressure actually do for you in practical terms?
Well, it's critical. If your plasma protein concentration drops significantly, say, due to severe protein malnutrition, or maybe a serious liver disease impairing protein synthesis fluid, stops returning efficiently from your tissues back into the circulation.
So it gets stuck out there.
Essentially, yes. It accumulates in the interstitial spaces, leading to visible swelling, a condition we call edema.
Like swollen ankles.
Exactly. It's a direct and often obvious consequence of this critical pressure imbalance. It dramatically demonstrates just how crucial these proteins are for maintaining the delicate fluid equilibrium throughout your entire body.
It's all about that pressure balance.
That leads us to an important question. How does fluid actually move so precisely between your blood and your surrounding tissues? It's this delicate, dynamic balance governed by 2 opposing forces.
Push and pull.
Essentially, yes. Hydrostatic pressure, which is basically the pressure exerted by the blood itself within the capillaries, acts to push fluid out of the capillaries and into the interstitial spaces. This delivers nutrients and oxygen to the cells.
Okay, pushing out.
Right. For example, in the arterioles, which are closer to the heart and have higher pressure, there's a net outward force of about 11 millimeters of mercury.
Got it.
Conversely, oncotic pressure, which is primarily exerted by the large plasma proteins because they can't easily cross the capillary walls, acts like a molecular sponge, pulling fluid back into the circulation from the tissues.
Pulling back in.
Okay, so in the venules, which are further along the capillary bed and where hydrostatic pressure has naturally dropped, there's a net inward force of about 9 millimeters of mercury.
So there's a net movement out at one end and back in at the other.
Precisely. This constant push and pull ensures proper fluid exchange, efficient nutrient delivery to your tissues, and effective clearing of waste products. It's a continuous dynamic dance.
Amazing. So how do these remarkable proteins get made in the first place and then, modified for their specific jobs?
Well, your liver truly is the primary synthesizer. It's responsible for producing roughly 70 to 80% of all plasma proteins.
The main factory.
Absolutely. This includes the heavy hitters like albumin, fibrinogen, transferrin, and most of the components that make up those vital complement and blood coagulation cascades we talked about.
Wow, 70–80% is huge. Are there exceptions?
There are a couple of prominent exceptions, yes. Von Willebrand factor, for instance, which is crucial for blood clotting, isn't synthesized in the liver, but rather in the vascular endothelium, the specialized lining of your blood vessels.
Ah, right at the site of action almost.
And the gamma globulins, which we'll discuss in more detail when we talk about immunity, are primarily synthesized in lymphocytes, key cells of your immune system.
Okay, so mostly liver, but some specialists elsewhere. What about modifications?
Right. Beyond their initial synthesis, most plasma proteins also undergo significant post-translational modifications. They are often covalently modified by the intricate addition of N or O-linked oligosaccharide chains.
Adding sugars, basically. Glycosylation.
Exactly, glycosylation. This adds complex functional sugar chains to their structure.
And what's the point of adding these sugars? Are they just decoration?
Oh, absolutely not. What's truly fascinating is that these sugar chains aren't just decorative, they fulfill a variety of critical functions. For example, the precise loss of terminal sialic acid residues, a type of sugar from these oligosaccharide chains, can actually accelerate the clearance of specific plasma glycoproteins from the circulation.
So it acts like a timer, or a signal for removal.
In a way, yes.
Yeah.
It acts as a crucial signal for their removal from the system. This highlights a sophisticated quality control mechanism, ensuring older or potentially damaged proteins are efficiently cleared. And to connect this to the bigger picture, for proteins destined for secretion, like the vast majority of plasma proteins, their journey from inside the cell to your bloodstream is a highly sophisticated multi-step process.
Right, the secretory pathway.
Exactly. It mirrors the complex cellular pathways we explored back in Chapter 49.
Okay, let's take a quick high-level tour then. How do these proteins get from inside the cell where they're made all the way out into the blood? Can you give us the grand tour of that journey?
Certainly. You can imagine these proteins are like meticulously crafted components moving along a finely tuned assembly line inside the cell.
Okay.
They're initially synthesized with a special passport, a signal sequence at their amino terminus that acts as a sort of zip code, targeting them specifically to the endoplasmic reticulum, the cell's internal network of membranes.
Got it, the ER.
As this leader sequence emerges from the ribosome, the protein-making machinery, it's recognized and guided by a protein complex called the signal recognition particle, or SRP.
Like an escort.
Exactly. This interaction pulls the emerging polypeptide chain through a channel into the lumen, the inner space, of the endoplasmic reticulum.
Okay, it's inside now.
Inside, its leader sequence, that passport, is precisely cleaved off by an associated enzyme called signal peptidase. The job is done.
Right.
The newly synthesized proteins then continue their journey, moving through the endoplasmic reticulum, then into the Golgi apparatus, which is essentially the cell's packaging and sorting center.
The post office of the cell.
A good way to put it. During this entire transit, they undergo various crucial post-translational modifications like further specific proteolysis, the addition of those glycosylation sugar chains we mentioned, maybe phosphorylation, all meticulously ensuring they're properly folded, stable, and fully functional.
Quality control checks along the way?
Absolutely. Ultimately, the mature proteins are neatly packaged into secretory vesicles, little membrane bubbles, and then released from the cell into the plasma, ready to perform their vital roles. It's an intricate journey of molecular maturation.
That really is a remarkable feat of cellular engineering. But once they're out there in the plasma, Are all these proteins, say, albumin molecules, identical across different people? Or is there some variation? And are they permanent or constantly being replaced?
That leads us to another important point. Are all these proteins identical across individuals? The answer is a clear no. Many plasma proteins exhibit what we call polymorphism.
Meaning multiple forms.
Exactly. They commonly exist in multiple distinct phenotypes or versions within the human population. This reflects the amazing genetic diversity within us, even right down at the level of our blood proteins.
Can you give some examples?
Sure. Classic examples include antitrypsin, haptoglobin, transferrin, ceruloplasmin, and of course the incredibly diverse immunoglobulins, which we'll get to.
So we're not all running exactly the same protein versions.
Not at all. And speaking of dynamics, a key insight is that each plasma protein has a characteristic half-life in the circulation.
How long it lasts.
Precisely. It's the specific time it takes for 50% of those particular protein molecules to be cleared or degraded from the bloodstream. This continuous turnover is absolutely essential.
Why? Why replace them?
Because it constantly replaces older, potentially damaged or less functional molecules with newly synthesized ones. This maintains a steady concentration and ensures the entire system is always refreshed and fully functional. It prevents accumulation of worn-out proteins.
Right. Keeps the machinery running optimally.
Exactly.
And here's where it gets really impactful clinically, right? In certain diseases, that half-life can change dramatically, acting as a diagnostic clue.
Absolutely. For instance, in conditions like Crohn's disease, which is a type of inflammatory bowel disease.
Right, regional ileitis.
Yes. It's characterized by significant protein loss into the bowel because the intestinal lining is inflamed and leaky. In severe cases, albumin's half-life can plummet from its normal 20 days down to just a single day.
20 days to 1 day, wow.
It's a massive drop. This severe reduction is a defining characteristic of what's called protein-losing gastroenteropathy. It provides a direct and striking example of how pathology profoundly impacts the fundamental dynamics and turnover of essential proteins within your body. It's a clear signal that something is seriously wrong with gut integrity. #Tag3. Albumin, the quintessential plasma protein.
Okay, let's zoom in on one particular protein, arguably the absolute rock star of plasma proteins, albumin. It truly dominates the scene, doesn't it?
It really does. Your liver synthesizes approximately 12 grams of albumin per day. That represents about 25% of its total protein synthesis capacity and an astonishing half of all the protein it secretes into your blood.
Half of its secreted protein is just albumin.
Just albumin. About 40% of the body's entire albumin pool circulates in the plasma, where it accounts for roughly 3/5, about 60% of your total plasma protein by weight. Typically, levels range from 3.4 to 4.7 grams per deciliter.
So it's incredibly abundant.
Extremely. And because of its sheer abundance and relatively high concentration, albumin contributes a remarkable 75 to 80% of the osmotic pressure, that oncotic pressure, of human plasma.
Wow, the vast majority.
Yes, making it the principal determinant of intravascular fluid balance and a silent guardian of your circulation, holding water inside the blood vessels.
Got it. And how is it made? Is it similar to the general pathway you described?
Yes, very similar. Like most other secreted proteins, albumin is initially synthesized as a pre-prop protein.
Meaning it has extra bits at the start.
Exactly. It begins with an extra signal peptide at its amino terminus, which is precisely removed as it enters the rough endoplasmic reticulum. Subsequently, a second smaller piece, a hexapeptide, is cleaved from the new N-terminus as it moves further through the secretory pathway. This ultimately yields the mature, functional protein.
So it gets trimmed down.
Correct. The mature human albumin consists of a single polypeptide chain, 585 amino acids in length. Its overall shape, its conformation is ellipsoidal, and it's incredibly stable. This stability comes from a total of 17 intrachain disulfide bonds that tightly hold its 3 functional domains together.
Those sulfur bridges really lock it in place.
They do. This robust structure is absolutely critical for its many, many roles.
And a major one, perhaps even the major role, is its incredible capacity for binding and transporting things, right? The molecular shuttle idea.
Absolutely. It truly is a molecular shuttle, carrying numerous vital ligands throughout your body, ensuring they get where they need to go safely and efficiently, without causing issues by being free in the plasma.
What kind of cargo does it carry?
Its specific cargo includes free fatty acids, FFAs, calcium ions, certain steroid hormones, bilirubin, a breakdown product of heme copper ions, and the amino acid tryptophan. It's a diverse array of crucial molecules that depend on albumin for their efficient transit.
It's like the blood's main taxi service for all sorts of important passengers.
That's a good way to think about it. And what's particularly impactful here is albumin's significant influence in medicine, especially pharmacology.
How so?
Well, many commonly used drugs, things like sulfonamides, penicillin G, decumarol, an anticoagulant, and even aspirin, bind directly to albumin in the bloodstream.
Okay.
This binding has profound pharmacologic implications. It affects how drugs are distributed throughout the body, their bioavailability, how much actually reaches the target tissues, and ultimately their efficacy. Albumin essentially acts as a temporary reservoir for the drug.
So it affects how much free drug is available to actually work.
Precisely. It influences how much free active drug is available at any given time. This binding can also impact drug interactions, potentially altering how different medications behave when taken together if they compete for the same binding sites on albumin.
That's really important for doctors to consider.
Absolutely. And on a therapeutic note, human albumin preparations themselves are widely used in medical settings. For example, in treating patients with severe burns or those suffering from hemorrhagic shock, administering albumin can rapidly restore circulating volume and maintain that crucial oncotic pressure.
So giving albumin directly can help pull fluid back into the blood vessels.
Correct. It's a key part of fluid resuscitation in those critical situations.
So what does this all mean when albumin production goes wrong? What are some clinical conditions related to albumin issues?
Well, there are rare genetic mutations that can profoundly impair albumin synthesis. This leads to a condition known as analbuminemia, where an individual's plasma is essentially completely devoid of albumin.
No albumin at all. What happens then? Massive edema?
Surprisingly, persons suffering from albuminemia often display only moderate edema.
Really? How is that possible if it provides 80% of the oncotic pressure?
It suggests the body has remarkable compensatory mechanisms to maintain fluid balance, likely involving increases in other plasma proteins, even without its primary osmotic player.
Yeah.
The body adapts.
Fascinating. What about more common situations?
Depressed synthesis of albumin also occurs in a variety of more common diseases, particularly those affecting the liver, since that's its primary synthesis site.
Like cirrhosis.
Exactly. Liver disease often manifests as a decrease in the ratio of albumin to the other plasma proteins, the globulins. This albumin globulin or Ag ratio is a common lab test.
Okay.
Furthermore, the synthesis of albumin decreases relatively early in conditions of severe protein malnutrition, such as kwashiorkor, which is often seen in developing countries.
Why does it decrease early?
It directly reflects the body's struggle to produce essential proteins when the necessary raw materials, the amino acids, are scarce. Albumin synthesis is prioritized downwards when resources are limited.
Okay, moving on from albumin, let's explore the body's really sophisticated acute phase response and its connection to inflammation. This is a critical protective mechanism that springs into action whenever your body faces threats like infection or injury.
A fundamental defense reaction.
The core idea here is that during inflammation or significant tissue damage, certain plasma proteins, which we call acute phase proteins, increase dramatically in concentration within your blood. They play a vital protective role in healing and defense.
Ramping up production of specific defenders.
Exactly. Key examples include C-reactive protein, or CRP, so named because it reacts with the C polysaccharide of pneumococci bacteria.
A historical naming convention.
Right. Other crucial players are ABON antiproteinase, which we met earlier as an inhibitor, haptoglobin, amino-acid glycoprotein, and fibrinogen, the clotting factor.
A whole suite of proteins gets upregulated.
And to truly grasp their power, consider this. CRP, for instance, actively stimulates the complement pathway, which is a crucial part of your immune defense, helping to flag pathogens for destruction.
Amplifying the immune response.
And ANTO1 antiproteinase acts like a dedicated damage control agent, neutralizing those dangerous proteases released during acute inflammation, thereby preventing widespread tissue damage. It's a remarkable rapid response system.
It's about both attacking the threat and protecting the host tissues.
Precisely.
And to elaborate on that, what's particularly useful here clinically is the diagnostic utility of CRP.
Yes, CRP tests are common.
Very common. During chronic inflammatory states, and also in patients with various types of cancer, the levels of these acute phase proteins can increase dramatically. CRP levels alone can potentially rise from 1.5-fold to as much as a 1,000-fold above the normal baseline levels.
1,000 times higher. That's incredible.
It's a huge increase. And because of this dramatic and generally reliable increase, CRP is widely used as a reliable biomarker of tissue injury, infection, and inflammation. It provides clinicians with a quick, objective snapshot of the body's inflammatory status.
A way to measure inflammation levels. And what actually orchestrates this powerful, acute phase response? How does the body signal this ramp up?
Cytokines.
Right. Expert, can you just simply explain what these are and how they trigger such a powerful internal response?
Certainly. Cytokines are essentially the immune system's communication network, tiny proteins that cells use to talk to each other and coordinate a defense.
Like molecular messages.
Precisely. They can act in an autocrine fashion, affecting the very cell that produced them, or in a paracrine fashion, signaling to nearby cells. A key insight here is that interleukin-1, IL-1, is recognized as a primary stimulator of acute phase protein synthesis by hepatocytes, your liver cells.
So IL-1 tells the liver, make more protective proteins.
Essentially, yes. And interleukin-6, IL-6, also participates significantly in this signaling. These are key signaling molecules orchestrating the liver's protein production response during inflammation, effectively telling the liver to ramp up its defensive protein factories.
Got it. IL-1 and IL-6 are major signals to the liver. Okay, here's where it gets truly intricate, I think. One of the primary targets of both IL-1 and IL-6 inside the cell is this factor called nuclear factor kappa B, or NFID. It acts like a master control switch, right? Activating a whole program of inflammation.
That's an excellent way to put it. NFID is a transcription factor, meaning it controls which genes get turned on or off. Normally, it resides in the cytosol, the main cell fluid, held inactive because it's bound to an inhibitory protein called IBA.
So it's kept on a leash normally.
Exactly. However, stimuli like inflammation, injury, or even radiation trigger the precise phosphorylation, adding a phosphate group to IBA. This phosphorylation acts as a tag, targeting IBA for ubiquitination and subsequent degradation by the proteasome, the cell's garbage disposal.
So the leash gets cut.
Precisely. Once freed from its inhibitory partner, the active NF can then translocate or move directly into the nucleus. Inside the nucleus, it binds to specific DNA sequences and stimulates the transcription of genes encoding many pro-inflammatory molecules, cytokines, chemokines, which attract other immune cells, growth factors, and cell adhesion molecules.
So it turns on a whole battery of inflammatory genes.
Yes. It's a critical on switch for inflammation, orchestrating a broad and rapid cellular response that mobilizes defenses and shapes the inflammatory environment. #Tag 5. Iron homeostasis. A tightly regulated dance.
Wow, okay. We've seen how essential these proteins are for defense, for fluid balance, but there's one particular element, iron, that requires its own incredibly complex and tightly regulated dance with specific proteins. It's like every single atom's accounted for.
It truly is a masterclass in biological regulation.
So let's now dive into how your body masters iron homeostasis, a process that is nothing short of vital. Iron is an absolutely essential constituent of so many human proteins. It plays fundamental roles in critical molecules like hemoglobin, carrying oxygen in your blood.
The most well-known role.
Myoglobin, storing oxygen in your muscles. The cytochrome P450 enzymes involved in detoxification. Numerous components of the electron transport chain, crucial for generating energy in the mitochondria.
ATP production.
And ribonucleotide reductase, which is fundamental for DNA synthesis. Without precise iron management, life as we know it simply couldn't exist.
It's central to so many core processes, and one crucial aspect here is how strictly the body conserves iron.
It holds onto it tightly.
Extremely tightly. A healthy adult typically loses only about 1 to 1.5 milligrams daily. That's less than 0.05% of their total body iron, which is usually around 3 to 4 grams.
Tiny losses compared to the total amount.
Exactly. This highlights its absolutely vital role and the body's elaborate, energy-intensive mechanisms designed to hold on to every bit of it. There's no dedicated physiological pathway for excreting excess iron, only for controlling absorption and recycling.
So regulation is mainly at the entry point and recycling.
Precisely. However, an important distinction is that premenopausal females typically experience higher losses, usually around 1.5 to 2 milligrams per day, largely due to menstruation. This naturally makes them more susceptible to iron deficiency.
Right. Regular blood loss increases the need.
Correct.
Now, a major part of this astonishing conservation is achieved through the body's highly efficient iron recycling system. orchestrated mainly by macrophages, right?
Yes, macrophages are key players here. Your red blood cells normally have a half-life of approximately 120 days.
Ah, 120, not 28. Got it.
Yes, 120 days, about 200 billion of them are catabolized, broken down every single day.
200 billion. That's mind-boggling.
It is. When these erythrocytes become senescent, old, or damaged, they are efficiently phagocytosed, literally eaten by macrophages, primarily found in the spleen and liver.
The cleanup crew.
Exactly. Within these macrophages, the hemoglobin-derived heme groups are oxidized by the enzyme heme oxygenase. This reaction releases carbon monoxide, which has its own signaling roles, and, critically, iron.
Okay, so iron is freed from the heme.
Yes. The liberated ferrous iron, F2+, is then exported from these macrophages back into the circulation by a specific transmembrane protein called ferroportin.
Ah, ferroportin again, the gatekeeper.
Precisely. So what's the bottom line here? Ferroportin is a central player, acting as a critical gatekeeper for both intestinal iron absorption and macrophage iron secretion, controlling its flow in and out of key cells.
Controlling the traffic.
Right. Once in the blood, this F2+ is oxidized to Fe3+ by ceruloplasmin, a copper-containing enzyme called a peroxidase, and then it's bound by transferrin for safe transport.
So copper is needed to load iron onto transferrin. Interesting link.
A very important link. This entire recycling system is incredibly efficient. About 25 milligrams of iron are recycled daily this way, which vastly reduces the need for new intestinal absorption, which averages only about 1 to 2 milligrams per day.
So recycling dwarfs absorption.
By a huge margin. It's essentially a closed-loop system, minimizing the need for external input and preventing iron loss.
Very smart design.
But that raises an important question. What happens if some hemoglobin escapes this meticulous recycling process? It's not 100% perfect.
Does some leak out?
Yes, approximately 10% of an erythrocyte's hemoglobin can escape into the circulation as free extracorpuscular hemoglobin during normal red cell turnover, or much more during hemolysis.
Okay, and that's a problem.
It can be. Hemoglobin is sufficiently small, around 65 kilodaltons, to potentially pass through the glomerulus of the kidney into the tubules. If it does, it tends to form damaging precipitates, potentially causing significant kidney injury.
Ah, clogging up the kidney filters.
Exactly. Haptoglobin, HP, another plasma glycoprotein, provides the ingenious solution. It specifically binds this free hemoglobin to form a large non-covalent complex, the HbHp complex.
How big is that complex?
It's much larger, 155 kilodaltons or more, depending on the haptoglobin type. The sheer size of this complex prevents its passage through the glomerulus.
Too big to filter out.
Right. This protects the kidney from harmful precipitates and crucially reduces the loss of valuable iron through the urine. Normal plasma contains enough haptoglobin to bind somewhere between 40 to 180 milligrams of hemoglobin per deciliter.
A decent capacity.
Yes. And human haptoglobin also exists in 3 common polymorphic forms, HP11, HP21, and HP22, reflecting genetic variations in the population.
Does it matter which form you have?
The different forms have slightly different properties, but the basic function is the same. And other plasma proteins like hemopexin and even albumin play complementary roles by binding free hemin rather than intact hemoglobin, further safeguarding the body from iron-related damage.
So multiple layers of protection. And to really understand haptoglobin's diagnostic power clinically, you mentioned low levels are significant.
Yes. In hemolytic anemias, conditions where red blood cells are constantly breaking down prematurely and releasing hemoglobin into the plasma, haptoglobin levels can plummet dramatically.
Why do they drop?
Because the HbHp complex, once formed, is rapidly cleared from circulation by macrophages. It has a much shorter half-life, only around 90 minutes, compared to free haptoglobin, which has a half-life of approximately 5 days.
Ah, so it gets used up quickly binding the excess hemoglobin.
Precisely. So a low haptoglobin level measured in a blood test strongly suggests an ongoing process of significant red blood cell destruction or hemolysis within the body. It's a key diagnostic indicator.
Got it. That makes sense. Now, what about getting iron into the body in the first place? Dietary absorption.
Right. Dietary iron absorption is another tightly regulated and complex process. Enterocytes, the specialized cells lining your small intestine, primarily absorb dietary iron in two main forms, as free ferrous iron, Fe2+, or as heme, the iron-containing part of animal proteins like hemoglobin and myoglobin.
So heme iron from meat and non-heme iron from plants or supplements.
Correct. The absorption of non-heme iron, which is primarily F2+O, occurs predominantly in the proximal duodenum, the first part of the small intestine, and it's exquisitely regulated based on the body's current iron needs.
How does it get across the cell membrane?
The crucial transfer of F2+ across the apical membrane, the side facing the gut lumen of these enterocytes, is mediated by a protein called divalent metal transporter 1, or DMT1.
DMT1, does it only transport iron?
Interestingly, no. It also surprisingly transports other essential and potentially toxic divalent metals like manganese, cobalt, zinc, copper, and even lead.
Wow, quite promiscuous.
It is, and since DMT1 is specific for F2+ VAC, any ferric iron, Fa3+, present in the diet must first be reduced to F2+ before it can be absorbed. This reduction can be done by ingested reducing agents like vitamin C, ascorbic acid, or by a brush border enzyme called a duodenal cytochrome B, D-site B.
So Fa plus needs converting first.
Yes. Once inside the enterocytes, the absorbed iron has two main fates. It can either be stored safely, bound to the iron storage protein ferritin.
The safe deposit box.
Right. Or it can be exported across the basolateral membrane, the side facing the bloodstream, into the circulation by that key protein, again, ferroportin.
Ferroportin, the exporter.
Exactly. And what's particularly ingenious here is the role of hepcidin. This is a copper-containing peroxidase, similar in function to ceruloplasmin in the blood.
Another copper connection.
Yes. Hepcidin oxidizes the Fe+ back to F3+ just before it's exported by ferroportin. This ensures that iron is in its less reactive, F3+ form, when it binds to transferrin in the plasma, minimizing the risk of oxidative damage.
So convert to Fe2+ to get in, convert back to Fe3+ to get out and travel safely.
You've got it. And any excess ferritin-bound iron that isn't exported from the enterocyte is simply disposed of when these intestinal cells are naturally sloughed off into the gut lumen every few days as part of normal gut turnover. This provides an additional passive layer of iron regulation, preventing overload if absorption exceeds immediate need.
A built-in disposal system for excess. Clever.
Very clever.
Okay, so once iron is absorbed, the body needs a way to store it safely, right? To prevent its toxic effects. That's where ferritin comes in.
Exactly. Your body can typically store up to 1 gram of iron, sometimes more, and the vast majority of this is bound safely within ferritin molecules. Think of ferritin as a molecular safe, a hollow spherical protein complex composed of 24 polypeptide subunits.
A protein cage.
A protein cage, precisely. It can encapsulate an astonishing 3,000 to 4,500 ferric Fe3+ atoms within its central core.
That's a huge storage capacity per molecule.
It is. These subunits come in two main types, H heavy and L light. The H subunit possesses peroxidase activity, which is essential for oxidizing F2+ to F3+ as it enters the ferritin core for storage. The L subunit is thought to play a role more in the nucleation and stabilization of that iron core. The ratio of H to L subunits can vary depending on the tissue type.
Tailored storage.
Yes. And a small amount of ferritin is also normally present in your plasma, typically in the range of 50 to 200 micrograms per deciliter. Crucially, this plasma ferritin level is generally proportionate to your total body iron stores.
So measuring plasma ferritin gives an idea of total body iron.
Correct. It makes plasma ferritin a very useful clinical biomarker for assessing overall iron status. Low levels indicate deficiency, high levels suggest overload. In conditions of severe iron overload, a partly degraded insoluble form of ferritin called hepcidin may also appear in tissues, visible under a microscope with iron stains, acting as a red flag for excessive accumulation.
Got it. Ferritin is the storage vault. Now, what about transport? Why is it so important to transport iron bound to something?
That leads to a critical question. Why is iron transported in a bound state and not just freely circulating? The answer lies in its inherent toxicity. Free iron, especially Fe2+A, is extremely dangerous.
Why dangerous?
Because it can readily catalyze the Fenton reaction. In this reaction, Fe2+ reacts with hydrogen peroxide, a normal byproduct of metabolism, to generate highly damaging reactive oxygen species, particularly the hydroxyl radical.
The hydroxyl radical. That sounds bad.
It is. It's transient but incredibly reactive. It can indiscriminately oxidize cellular macromolecules, lipids, proteins, DNA, causing significant oxidative stress and tissue damage throughout the body.
So free iron is like a road catalyst for damage.
Exactly. To protect against this potential havoc, living organisms employ specialized storage proteins like ferritin and dedicated transport proteins. In humans, the vast majority of iron in the circulation, specifically F3 plus, say, is transported bound to transferrin.
The iron taxi.
The iron taxi, yes. It's a glycoprotein synthesized primarily by the liver. It circulates as a 1-globulin with a molecular mass of approximately 76 kilodaltons. Crucially, it contains 2 high-affinity binding sites specifically for Fe3 plus C, ensuring iron is always safely chaperoned during transit.
2 seeds per taxi.
Right. And interestingly, the glycosylation, the sugar chains attached of transferrin, can be impaired in certain congenital disorders of glycosylation or, notably, in chronic alcoholism. In the latter case, the presence of carbohydrate-deficient transferrin, CDT, serves as a valuable biomarker for detecting chronic alcohol abuse.
Ah, another biomarker connection.
Yes. The concentration of transferrin in plasma is typically about 300 milligrams per deciliter. This amount is sufficient to carry around 300 micrograms of iron per deciliter. This maximum carrying capacity is referred to as the total iron binding capacity, or TIBC.
Okay, TIBC is how much iron the transferrin could carry.
Correct. Normally, only about 30% of transferrin's iron binding sites are actually occupied. This percentage, the transferrin saturation, is another key clinical measure. It can decrease significantly, often to less than 16% during severe iron deficiency, simply because there's less iron available to bind. Conversely, it may increase to more than 45% in conditions of iron overload. These measurements provide crucial diagnostic insights into the body's iron balance.
So saturation tells you how full the taxis are.
Exactly.
Okay, so transferrin carries the iron safely in the blood. To really grasp how that iron then gets into your cells from the bloodstream, let's explore this highly sophisticated and efficient recycling process known as the transferrin cycle. Expert, could you walk us through how this molecular dance unfolds?
Absolutely. Think of it as a meticulously choreographed delivery and return system. It starts when holotransferrin that's transferrin bound to iron, TFE, first binds to a specific receptor on the cell surface called transferrin receptor 1, or TFR1.
The cell puts out a receptor to catch the iron taxi.
Precisely. These receptors are often concentrated in specialized areas of the cell membrane called clathrin-coated pits, which are designed for internalizing molecules. This entire complex TFE, bound to TFR1, is then internalized by a process called receptor-mediated endocytosis, forming little vesicles called early endosomes inside the cell.
Okay, the whole package gets brought inside.
Yes. These early endosomes then mature into late endosomes. A key feature of late endosomes is that they become progressively more acidic. Their internal pH drops significantly.
Why the drop in pH?
This low pH environment within the late endosome is absolutely crucial because it causes the release of F3+ from transferrin. The acidic conditions weaken the bond.
Ah, the acid pops the iron off.
Effectively, yes. The released ferric iron, Fe3+, is then rapidly converted to its ferrous form, Fe2+, by a ferroreductase enzyme called STEAP3, located within the endosome membrane. This F2+ is then transported out of the endosome and into the cytosol, the main cell fluid, by DMT1, that same divalent metal transporter we encountered in the gut.
DMT1 again. Busy protein.
Very busy. Now, what's truly clever about this system is what happens next. The iron-free transferrin, now called apotransferrin or ApoTef, remains bound to the TFR1 receptor even after releasing its iron.
It doesn't get degraded.
No. The entire ApoTF-TFR1 complex is efficiently recycled back to the cell surface within the endosomal vesicle. Once it reaches the cell surface, the neutral pH of the extracellular environment causes the ApoTF to dissociate from the receptor. The ApoTF re-enters the plasma, ready to pick up more iron, and the TFR1 receptor is ready to bind another holotransferrin molecule, completing the cycle.
Wow. Elegant recycling of both the carrier and the receptor.
Extremely elegant and efficient. And while TFR1 is found on most dividing cells needing iron, there's a related receptor, TFR2, which is primarily found on hepatocytes, liver cells, and intestinal Krebs cells. TFR2 has a much lower affinity for transferrin, suggesting it functions more as an iron sensor, monitoring body iron levels rather than being a primary iron importer like TFR1. It plays a more nuanced role in overall iron regulation.
Okay, a different receptor for sensing rather than just uptake. Now, what's truly fascinating is how ceruloplasmin, that copper-containing enzyme we mentioned, ties directly into this precise iron cycle. Expert, what's its crucial role again?
Ceruloplasmin is a relatively large protein, a 160 kilodalton A2 globulin synthesized in the liver. It is the major copper-containing protein in plasma. Its key role in iron metabolism is to oxidize F2+ to F3+ seru.
Why is that oxidation step so important again?
It's critical because transferrin can only bind F3 plus effectively. Remember that iron release from macrophages via ferroportin is in the F2 plus state. Ceruloplasmin circulating in the plasma oxidizes this F2 plus to Fe3 plus sera, making it available for binding by transferrin and subsequent transport via the transferrin cycle. Without ceruloplasmin's peroxidase activity, iron recycling from macrophages would be severely impaired.
So it preps the iron release from recycling to get back onto the taxi.
Exactly. Ceruloplasmin carries out this vital oxidation using 6 catalytically essential copper atoms embedded within its structure, demonstrating again that intricate and often surprising link between copper and iron metabolism.
Okay, so copper is essential for proper iron transport. And when ceruloplasmin is deficient, that obviously messes up iron homeostasis, leading to health issues.
Yes, significantly. Ceruloplasmin deficiencies can arise from genetic causes or simply from a lack of dietary copper, which is an essential micronutrient. When there isn't enough functional ceruloplasmin, the body's ability to efficiently recycle FP2 plus emerging from macrophages for transport becomes impaired.
So iron gets stuck.
It can lead to iron accumulation in various tissues, particularly the liver and brain, while paradoxically potentially causing anemia because iron isn't efficiently delivered to red blood cell precursors. It's a complex picture of miscompartmentalized iron. For instance, individuals suffering from hypoceruloplasmia, a genetically inherited condition where ceruloplasmin levels are about 50% of normal, generally show no major clinical abnormalities, surprisingly. The remaining activity seems sufficient. However, genetic mutations that completely abolish the peroxidase activity of ceruloplasmin, a condition called aceruloplasminemia, can have severe physiologic consequences. If left untreated, the progressive accumulation of iron in specific tissues like pancreatic islet cells and the basal ganglia of the brain can lead to the development of insulin-dependent diabetes and significant neurologic degeneration.
What kind of neurological problems?
Manifesting as dementia, dysarthria, difficulty speaking, and dystonia, involuntary muscle contractions. It's a serious neurodegenerative disorder linked directly to iron misregulation due to ceruloplasmin loss.
Wow.
And here's where it gets really interesting and somewhat paradoxical: Wilson disease.
Right, you mentioned this earlier. Copper overload, but low ceruloplasmin.
Exactly. It's a condition that highlights the delicate interplay between copper and ceruloplasmin synthesis. Wilson disease is caused by mutations in the gene for a binding P-type ATPase called the ATP7B protein. This protein is normally responsible for loading copper onto apoceruloplasmin, ceruloplasmin without copper, inside liver cells and for excreting excess copper into the bile.
Right.
The mutation essentially blocks both these functions. This causes copper to dangerously accumulate within vital organs like the liver, brain, kidneys, and red blood cells. But paradoxically, these rising levels of copper inside the liver cell interfere with the proper incorporation of copper into newly synthesized ceruloplasmin polypeptides.
So the excess copper prevents proper ceruloplasmin formation.
Correct. This leads to a significant fall in functional ceruloplasmin levels in the plasma, despite the systemic copper overload. Untreated, patients can develop hemolytic anemia, chronic liver disease like cirrhosis, and severe neurologic symptoms due to copper accumulation in the basal ganglia.
A very strange situation. Is it treatable?
The good news is that Wilson disease is treatable, often dramatically improving patient outcomes. Treatment involves carefully limiting dietary copper intake and using chelating agents like penicillamine or trientine, which bind excess copper and help remove it from the body via urine. Zinc therapy is also used, as it induces metallothionein in the gut, which binds copper and prevents its absorption.
Okay, so manageable if caught and treated.
Yes, management is key.
This all raises an important question. With all this complexity, how do individual cells know exactly how much iron they need? It can't just be a free-for-all, right? There must be internal regulation, too.
Absolutely. There's a brilliant reciprocal regulation at play within each cell to manage its own intracellular iron homeostasis. Changes in the intracellular iron level profoundly influence the synthesis rates of both transferrin receptor 1, TFR1, the iron importer, and ferritin, the iron storage protein.
Reciprocal how?
When cellular iron is low, the cell needs to import more. So the rate of TFR1 synthesis increases. It puts out more iron antennas to catch transferrin. At the very same time, ferritin synthesis declines because there's less iron to store in the iron storage lockers, and the cell wants to prioritize using the iron it has.
More import, less storage when low. Makes sense.
Exactly. And the precise opposite occurs when intracellular iron is abundant, indicating the tissue's immediate needs have been met. TFR1 synthesis goes down and ferritin synthesis ramps up to safely store the excess. It's a beautifully precise, self-adjusting system at the cellular level.
Okay, but how does the cell sense the iron level and adjust protein synthesis accordingly? What's the molecular mechanism?
This elegant control is primarily exerted through specialized proteins called iron regulatory proteins, or IRPs, specifically IRP1 and IRP2. These are actually cytoplasmic isoforms of the well-known TCA cycle enzyme aconitase.
Aconitase, doing double duty.
Indeed. These IRPs have the ability to bind to specific hairpin loop structures in messenger RNAs, mRNAs, called iron response elements, or IREs.
IREs. Okay, where are they found?
They are strategically located in either the 5 or 3-foot untranslated regions, UTRs, the non-coding ends of the mRNAs that code for ferritin and TFR1, respectively.
Ah, so they control the fate of the mRNA messages for these proteins.
Precisely. Here's how it works. When cellular iron is low, the IRPs do not have iron bound to them. In this iron-free state, IRP binds to the IRE located in the 3-foot UTR of the TFR1 mRNA. This binding actually stabilizes the mRNA, preventing its degradation and thereby increasing TFR1 synthesis so the cell can import more iron.
Okay, low iron IRP binds TFR1 mRNA, more TFR1 protein.
Correct. At the same time, iron-free IRP binds to the IRE located in the 5-foot UTR of the ferritin mRNA. Binding here physically blocked the ribosome from initiating translation, so no ferritin protein is made, ensuring that every bit of available iron is used functionally rather than being stored away.
Low iron IRP binds ferritin mRNA, no ferritin protein.
Got it. Now, conversely, when intracellular iron levels are high, Fe2+ binds directly to the IRPs. This binding triggers the assembly of a 4Fe4S iron sulfur cluster within the IRP molecule, causing a conformational change.
The IRP changes shape when iron binds.
Yes. This structural change causes the IRP to dissociate, to let go from the IREs on both mRNAs. Once the ferritin mRNA is free of IRP in its 5-foot UTR, it becomes available for translation, so ferritin is rapidly synthesized to store the excess iron.
High IRP releases ferritin mRNA, more ferritin protein.
Correct. Concurrently, when IRP dissociates from the 3-foot UTR of the TFR1 mRNA, that mRNA becomes susceptible to rapid degradation by cellular enzymes. This effectively slows down TFR1 synthesis as less iron import is needed.
High iron IRP releases TFR1 mRNA, less TFR1 protein. Wow.
It's a sophisticated molecular dance of regulation, perfectly coordinating iron uptake and storage based on the cell's immediate iron status.
So what's the core takeaway here? It's truly a genius system. Your cells have this built-in iron thermostat that constantly adjusts how much iron they import and store, ensuring perfect balance right down to the single atom.
That's a great way to visualize it.
When iron is scarce, your cells put out more iron antennas, TFR1, to grab all available iron and simultaneously shut down the iron storage loggers, ferritin, to ensure every bit is used. When iron is plentiful, they do the exact reverse, build more storage and reduce import. It's a precise molecular switch, ensuring cellular iron balance is always maintained.
Beautifully summarized. But that's intracellular regulation. What about the whole body?
Right. How is the systemic balance controlled?
What's particularly fascinating here is the discovery of the peptide hormone hepcidin. This small 25 amino acid peptide, synthesized primarily in the liver, is now recognized as the chief regulator of systemic iron homeostasis. It essentially acts as the master switch, controlling iron levels throughout the entire body, coordinating absorption, recycling, and storage.
Hepcidin, the master regulator. How does it work?
Its primary mechanism of action is to bind directly to ferritin, that cellular iron exporter protein we keep encountering.
On gut cells and macrophages.
Exactly. Hepcidin binding to ferritin triggers ferritin's internalization into the cell and its subsequent degradation.
So hepcidin gets rid of the iron exit doors.
Precisely. This has profound consequences. The resulting decrease in ferritin on cell surfaces produces what's called a mucosal block, which significantly lowers iron absorption from your diet in the intestine. It also depresses the recycling of iron liberated from red blood cell turnover by macrophages.
Okay, so it reduces iron getting in from the diet and iron getting out from recycling.
Correct. Together, these actions result in a crucial reduction in circulating iron levels, a state known as hypophoремia. Hepcidin even impacts placental iron transfer during pregnancy, ensuring iron distribution to the fetus is also tightly regulated.
Wow, powerful little peptide. How is its production controlled?
Crucially, when plasma iron levels are high, the synthesis of hepcidin by the liver increases. This creates a powerful negative feedback loop. High iron leads to more hepcidin, which then reduces iron absorption and recycling, thereby bringing circulating iron levels back down into the normal range.
A self-correcting system.
Exactly.
But how exactly does the liver sense the body's iron levels to regulate hepcidin production? It must have sensors too, right?
It does. Liver cells monitor body iron status using one of two sophisticated iron sensing complexes located on their surface. These complexes typically consist of a homodimer of either TFR1 or TFR2, bound to a third crucial transmembrane protein called HFE.
HFE. That sounds familiar. Hemochromatosis gene.
Precisely. HFE protein is a major histocompatibility MHC class 1-like molecule that normally binds to TFR1. When body iron is abundant and levels of iron-bound transferrin, TFA, in the plasma are high, TFE competes with HFE and displaces it from TFR1.
Okay, high iron kicks HFE off TFR1.
Yes. The displaced HFE protein is then thought to bind to TFR2, forming a complex that can be further stabilized by association with TFA. Formation of this HFE TFR2 TFE complex triggers an intracellular signaling cascade, specifically activating the ARKEK pathway.
A common signaling pathway.
Yes. This pathway ultimately leads to increased expression of the HAMP gene, which is the gene that encodes hepcidin.
So high iron HFE moves to TFR2 signaling cascade, more hepcidin produced.
That's the essence of it. And it's profoundly significant that mutations in the gene encoding HFE protein are the most common cause of hereditary hemochromatosis, directly linking this sophisticated sensing mechanism to the devastating consequences of systemic iron overload in affected individuals. Their sensor is broken.
Wow, that really connects the dots. Are there other signals influencing hepcidin?
Yes, indeed. Bone morphogenetic proteins, BMPs, particularly BMP6, also significantly influence hepcidin expression. They act via their own cell surface receptor, BMPR, and a co-receptor called hemojuvelin, HJV.
More receptors involved.
Activation of this BMPR HJV complex triggers the phosphorylation of intracellular signaling proteins called SMADs. These activated SMADs then move to the nucleus and stimulate the transcription of the HAMP gene, leading to increased hepcidin production, especially in response to increased liver iron stores.
Okay, so BMPs add another layer of iron sensing input.
Correct.
Now, this intricate regulation also means that things like inflammation or the body's need for red blood cells must significantly impact hepcidin levels too, right? Overriding other signals sometimes.
Absolutely. Inflammatory and erythropoietic red blood cell production signals are powerful regulators of hepcidin. For example, during an inflammatory response, pro-inflammatory cytokines like interleukin-6, IL-6 bind to their specific receptors on liver cells. This activates another signaling pathway called the JAK-STAT pathway, which in turn strongly induces hepcidin expression.
Inflammation makes more hepcidin. Why would it do that?
This is why the anemia of inflammation, or anemia of chronic disease, often accompanies chronic inflammatory states. The body's inflammatory response actually reduces iron availability by increasing hepcidin. This restricts iron absorption and recycling, likely as a defense mechanism to withhold iron from invading pathogens that rely on it for growth.
Ah, starving the invaders of iron.
Yeah.
Makes sense, but causes anemia as a side effect.
Precisely. Conversely, hepcidin expression dramatically decreases during hypoxia, a state of low oxygen. This reduction is mediated partly by erythropoietin, EPO, the hormone stimulating red cell production, and also by hypoxia-inducible transcription factors, HIFs. This ensures more iron is made available for hemoglobin synthesis when oxygen levels are low and more red blood cells are needed.
Makes sense. Need more red cells, need more iron.
And in conditions like beta thalassemia, where red blood cell production is ineffective but highly active, hepcidin expression is also strongly inhibited. This is driven by factors like growth differentiation factor 15, GDF15, and twisted gastrulation 1, TWSG1, which are secreted by the developing erythroblasts themselves. Again, this ensures maximum iron availability for attempting red blood cell production, even though it can contribute to iron overload in these patients.
So many layers of control. Incredible.
It is. And one crucial aspect of these regulatory mechanisms is their direct relationship to major global health concerns. Iron deficiency and the resulting anemia are extremely common worldwide, particularly in developing countries, affecting millions and millions of people, especially women and children.
But what are the main causes?
Major causes include dietary deficiency, lack of iron-rich foods, malabsorption issues in the gut, chronic or acute gastrointestinal bleeding, like from ulcers or parasites, and episodic blood loss, such as heavy menstruation. Persistent iron deficiency leads to the progressive depletion of the body's iron stores, first the storage iron and ferritin, then functional iron. If the level of transferrin saturation falls significantly, often to 20% or below, hemoglobin synthesis becomes significantly impaired. This results in what's known as iron-deficient erythropoiesis, making red cells without enough hemoglobin.
So the factory slows down.
Exactly. Over time, hemoglobin levels in the blood will gradually fall, leading to full-blown iron deficiency anemia. Clinically, this typically manifests as a hypochromic microcytic blood picture.
Meaning the red cells are pale and small.
Correct.
Yeah.
Paler hypochromic because they lack hemoglobin, and smaller microcytic. This is often accompanied by debilitating symptoms like fatigue, pallor, paleness, shortness of breath, and reduced exercise capacity.
Common symptom.
Very common.
So what does this all mean for diagnosis in a clinical setting? What are the key biomarkers doctors look for?
During iron-deficient anemia, red blood cells exhibit increased levels of surface transferrin receptor 1, TFR1, as the cells desperately try to grab more iron. There are also deficits in the ferrochelatase catalyzed incorporation of iron into protoporphyrin IX, the final step in heme synthesis. Crucially, partial proteolysis of these increased cell surface transferrin receptors releases elevated quantities of soluble transferrin receptor, STFR protein, into the plasma.
STFR, another useful marker.
Yes. This increase in STFR, along with the accumulation of unused red cell protoporphyrin, serve as key diagnostic biomarkers, specifically for iron deficiency anemia. This is particularly useful because chronic inflammation, which can also cause anemia by altering hepcidin levels, does not significantly affect the level of erythrocyte transferrin receptors in the same way.
So STFR helps distinguish iron deficiency anemia from anemia of chronic disease.
Generally, yes. STFR levels tend to remain relatively normal in anemia of chronic disease, but are elevated in iron deficiency. Key laboratory tests for assessing comprehensive iron status, therefore, include serum ferritin, for stores, TIBC, total transferrin, serum iron transferrin saturation, iron level relative to TIBC, red blood cell protoporphyrin, STFR, and careful examination of red blood cell morphology under the microscope.
A whole panel of tests to get the full picture.
Correct. These specific biomarkers allow clinicians to precisely detect and monitor iron deficiency anemia, differentiating it accurately from other types of anemia, which is critical for choosing the right treatment.
Makes sense. Now, on the absolute opposite end of the spectrum, what about iron overload? Hereditary hemochromatosis.
Right. Hereditary hemochromatosis, or iron overload, is characterized by hemosiderosis, that pathological accumulation of stainable concentrations of iron in various tissues, leading eventually to organ damage and failure if untreated.
What causes this overload?
It can arise from several causes. The most common is hereditary hemochromatosis, which is primarily due to hereditary hyperabsorption of iron by the intestines. This is most commonly caused by those mutations in the HFE gene we discussed, the iron sensor gene. Less frequently, it can be caused by mutations in genes for hepcidin itself, TFR2, hemojuvelin, HJV, or even ferroportin, leading to iron trapped in macrophages or unregulated release.
So, genetic defects in the control system.
Exactly. Secondary iron overload, on the other hand, is usually associated with conditions of ineffective erythropoiesis, like thalassemia syndromes, where the body tries to make many red cells but fails, signaling for more iron absorption inappropriately. It can also result from repeated blood transfusions. Each unit contains significant iron, parenteral iron therapy, iron injections, or historically massive dietary iron overload, such as Bantu siderosis, seen in African populations consuming traditional beer brewed in iron pots.
Wow, beer causing iron overload.
Historically, yes, due to the massive iron content. Additionally, miscellaneous conditions like alcoholic liver disease, non-alcoholic steatohepatitis, NH, and hepatitis C infection can also be associated with secondary iron overload, demonstrating the broad impact of liver health and other factors on iron dysregulation. The core pathology in the hereditary forms, however, is that inappropriate hyperabsorption of iron by the intestines due to faulty regulation. #Tag6. Serum inhibitors preventing indiscriminate proteolysis.
Okay, let's shift gears now to another crucial set of protective proteins in the plasma, the serum inhibitors. These play a vital role in preventing indiscriminate proteolysis, basically, stopping protein-cutting enzymes from running wild.
A critical balancing act.
Proteosis, these enzymes that break down proteins, are absolutely essential participants in countless physiological functions, right? Things like tissue remodeling during growth or healing, the complex process of blood clotting, the precise elimination of old or diseased cells, and the crucial destruction of invading pathogens. They are really the body's demolition and recycling crew.
Essential but potentially dangerous tools.
Exactly. So that leads to the important question. If proteins are so crucial and powerful, what stops them from just attacking healthy tissue all the time?
Right. If left unchecked, these powerful proteolytic enzymes, whether they're secreted as part of a defense mechanism or released into the blood due to tissue damage, can cause significant and widespread harm.
Like friendly fire causing collateral damage.
Precisely. The body's protection from this indiscriminate proteolysis involves a battery of specialized serum proteins inhibitors that specifically recognize, bind, and neutralize protease activity, limiting their scope of action. It's a crucial regulatory system to ensure their power is harnessed appropriately, not unleashed destructively.
Okay, who are the main players in this inhibitor team?
One of the most important is A1 antiproteinase, which was formerly known and perhaps more widely recognized as A1 antitrypsin.
Ah, all-1 antitrypsin linked to emphysema.
Exactly. This 394 residue glycoprotein is the principal serine protease inhibitor, or SERPIN, in human plasma. It works by inactivating various serine proteases enzymes like trypsin, chymotrypsin, and crucially neutrophil elastase by forming a stable, essentially irreversible covalent complex with them, effectively neutralizing their activity.
It traps them.
It traps them. I1 antiproteinase constitutes more than 90% of the I1 globulin fraction measured in plasma electrophoresis and is primarily synthesized by hepatocytes in your liver and also by macrophages, those key immune cells.
Okay, and you mentioned genetic diversity earlier.
Yes, there's fascinating genetic diversity here too. There are at least 75 known polymorphic forms designated by the pi protease inhibitor system. The major genotype found in most individuals of European descent is PMM.
And deficiency is the problem.
Yes. Here's where its deficiency becomes clinically significant. A severe deficiency in functional I1 antiproteinase plays a significant causal role in about 5% of all emphysema cases. This is particularly true for individuals with the PiZ genotype, who synthesize a defective PiZ protein that gets stuck in the liver and isn't secreted properly, or in PiSZ heterozygotes. Both of these groups secrete substantially lower levels of functional SERPIN into the plasma than normal PiMM individuals.
So their lungs lack protection against elastase.
Exactly. Neutrophil elastase, released during inflammation in the lungs, can then progressively destroy the elastic tissue, leading to emphysema. Their lungs are profoundly more vulnerable to this proteolytic damage.
And what about lifestyle factors? Smoking.
To elaborate on that, what's particularly insightful here is the direct link to lifestyle, specifically smoking. The oxidation of a key methionine residue, MET358, located precisely in the reactive center loop, the protease-binding domain of A1 antiproteinase, renders the inhibitor inactive. It can no longer bind and neutralize elastase.
Oxidation breaks the trap.
Correct. Individuals who use tobacco products or are regularly exposed to smoke from burning fossil fuels or other sources of oxidants generate high levels of reactive oxygen species in their lungs. These oxidants are especially prone to causing this specific oxidation of MET358 in I1 antiproteinase. This accelerates the development of emphysema, even in people with normal PMM genotypes, because their crucial protective inhibitor is rendered functionally ineffective by the oxidative stress.
So smoking effectively causes a functional deficiency.
In essence, yes. And to counteract this in specific patient populations with genetic deficiency, intravenous administration of purified I1 antiproteinase, known as augmentation therapy, has been used as an adjunct treatment for emphysema patients who exhibit this severe deficiency, providing the missing protective factor directly to the lungs.
Replacing the missing shield.
Yes. Furthermore, this deficiency, specifically the PiZ phenotype, is also linked to I1 antitrypsin deficiency liver disease. This is a form of cirrhosis that affects certain individuals. In these cases, the specific mutation underlying the PiZ protein, GLU342 to lysine, causes the protein to misfold and aggregate abnormally within the endoplasmic reticulum of hepatic cells. This accumulation triggers cellular stress and inflammation, leading eventually to liver damage and cirrhosis.
So the same defect causes lung problems by deficiency and liver problems by accumulation. Wow.
Exactly. It's a dual pathology from a single genetic defect.
Okay, who else is on the inhibitor team? Is there a broader spectrum player?
Yes, another critical protease inhibitor, one that acts as a true pan-protease inhibitor and even has other roles like cytokine transport, is ABA2 macroglobulin.
ABA2 macroglobulin.
Yeah.
Sounds big.
It is. It comprises a substantial 8 to 10% of the total plasma protein in humans. It's a large, homotetrameric glycoprotein, four identical subunits synthesized by various cells, including monocytes, hepatocytes, and astrocytes. Interestingly, it's a member of the thioester plasma protein family, which surprisingly also includes complement proteins C3 and C4, hinting at some evolutionary relationships.
A theoester. That sounds reactive. How does it work?
It has an ingenious mechanism. A2 macroglobulin employs what's often described as a venous flytrap mechanism for neutralizing proteases from virtually all catalytic classes.
Venous flytrap. Intriguing.
It possesses a unique, flexible region called the bait domain, about 35 residues long. When an attacking protease cleaves within this bait domain, it doesn't destroy the A2 macroglobulin. Instead, the cleavage triggers a massive conformational change in the entire A2 macroglobulin molecule.
The trap springs shut.
Exactly. The molecule collapses around the protease, physically enveloping and trapping it within the structure. Then, a reactive internal cyclic thioester bond within A2 macroglobulin becomes exposed and covalently links the inhibitor to the trapped protease, forming a stable complex and firmly locking the protease inside.
Covalently links them. That's a strong bond.
Very strong. And this conformational change does something else too. It exposes a specific recognition sequence on the surface of the A2 microglobulin molecule. This sequence is recognized by specific cell surface receptors, particularly on macrophages. This leads to the rapid and efficient clearance of the entire A2 macroglobulin protease complex from the plasma, ensuring these dangerous neutralized proteases are removed from circulation before they can cause trouble.
So it traps, neutralizes, and flags for disposal. Very efficient.
Extremely efficient.
And you mentioned other roles, cytokine transport.
Yes. Building on that, A2 macroglobulin isn't just a simple inhibitor, it's also a sophisticated delivery system for certain signaling molecules. In addition to serving as the plasma's predominant broad-spectrum protease inhibitor, it also binds and transports approximately 10% of the plasma zinc, an important trace element. And crucially, it binds various cytokines, including important growth factors like platelet-derived growth factor, PDGF, and transforming growth factor beta, TGF.
Why bind cytokines?
This binding likely serves to protect these potent signaling molecules from degradation and perhaps routes these vital bound effectors toward particular tissues or target cells. Once the complex is taken up by the target cell via receptor-mediated endocytosis, the cytokine likely dissociates within the endosomal compartment, thereby freeing it to exert its specific modulatory effects on cell growth, differentiation, or function. It's truly a multifunctional molecular actor in the plasma.
Inhibitor, transporter, delivery system. Very versatile. Hashtag Tech7. Amyloidosis protein deposition in tissues. Now let's turn our attention, perhaps briefly, to a more unfortunate consequence when proteins go seriously awry. Amyloidosis. This involves the pathological deposition of abnormal proteins in tissues, causing damage.
Yes, a group of diseases characterized by protein misfolding and aggregation.
Amyloidosis refers to the impairment of tissue function that results from the accumulation of these abnormal, insoluble protein aggregates in the interstitial spaces, the areas between cells,
disrupting normal tissue architecture and function.
And the term amyloidosis itself is actually a bit of a historical misnomer, isn't it?
It is. It was originally thought, based on early staining properties with iodine, that these fibrils were starch-like, hence amyloid, from Latin amylum, starch.
But they're not starch at all.
No, not at all. We now know that the characteristic amyloid fibrils are actually made up primarily of proteolytic fragments derived from various precursor plasma proteins. A key feature is that these fragments adopt an abnormal conformation extremely rich in mole-pleated sheets.
That beta sheet structure is key.
Yes, this specific protein folding structure makes them highly stable, insoluble, resistant to normal proteolytic degradation, and prone to aggregate into these characteristic fibrils that deposit in tissues. These pathological aggregates generally also contain a non-fibrillar component called the P component, which is derived from a normal circulating plasma protein called serum amyloid P component, SAP, which is structurally related to C-reactive protein.
Okay, so misfolded protein fragments forming insoluble fibrils. What are the main types?
Well, the most common type overall is primary amyloidosis, often called AL amyloidosis. So what's the clinical relevance here? This type is often caused by an underlying monoclonal plasma cell disorder, essentially a type of blood cancer like multiple myeloma, where plasma cells produce excessive amounts of abnormal immunoglobulin light chains. Fragments of these light chains then misfold and aggregate, forming the amyloid deposits.
So it's linked to antibody production gone wrong.
In this case, yes. Then there's secondary amyloidosis, or AA amyloidosis. This results from an accumulation of fragments derived from a different precursor protein called serum amyloid A,
Right.
SAA.
SAA. Is that an acute phase protein?
Yes, exactly. SAA is an acute phase protein whose levels rise dramatically during inflammation. AA amyloidosis is typically associated with chronic infections like tuberculosis or osteomyelitis historically, or chronic inflammatory conditions like rheumatoid arthritis, inflammatory bowel disease, or certain cancers. In these conditions, sustained high levels of inflammatory cytokines stimulate the liver to continuously overproduce SAA. This leads to increased levels of proteolytic degradation products of SAA that then misfold and accumulate as amyloid fibrils, particularly in the kidneys, liver, and spleen.
So chronic inflammation can lead to this type.
It's a major risk factor. Then there are various forms of familial or hereditary amyloidosis. These result from the accumulation of mutated forms of certain normal plasma proteins, such as transferrin TTR, which normally transports thyroid hormone and vitamin A. Over 80 different mutations in the TTR gene have been identified that cause it to misfold and form amyloid deposits, leading to various syndromes affecting the nerves, heart, or other organs. Fibrinogen anachain and apolipoprotein A1 are other examples of proteins whose mutations can cause hereditary amyloidosis.
So inherited mutations can predispose to amyloid.
Correct.
And that raises an important question. Why does long-term kidney dialysis sometimes lead to a specific type of amyloidosis?
Yes, dialysis-related amyloidosis. This is a significant clinical concern for patients on long-term hemodialysis. The precursor protein here is V2 microglobulin, V2M, a small protein normally found on the surface of most cells and present at low levels in plasma. Healthy kidneys filter out V2M, but standard dialysis membranes are unable to clear it effectively from the blood.
So it builds up in dialysis patients.
Exactly. Over years of dialysis, beta-LIM accumulates to very high levels in the blood. This increases the propensity for it to aggregate and form amyloid fibrils, which typically deposit in bones and joints, osteoarticular tissues, leading to conditions like carpal tunnel syndrome, destructive arthropathy, and bone cysts.
A complication of the treatment itself.
Unfortunately, yes, though newer dialysis membranes are better at clearing beta-LIM. And finally, it's also worth noting that a different type of amyloid, formed from the amyloid beta peptide, AA, is critically implicated as a key pathological feature in Alzheimer's disease, depositing as plaques in the brain. This highlights the diverse range of devastating conditions, including neurodegenerative diseases, where protein misfolding and aggregation into amyloid fibrils can lead to severe pathology, affecting millions worldwide.
A common pathological mechanism in diverse diseases. #Tag 8. Plasma immunoglobulins, the adaptive immune defense. Okay, finally, let's dive into the truly fascinating world of plasma immunoglobulins, the incredibly sophisticated adaptive immune defense system that protects us from a mind-boggling universe of pathogens and other threats.
The heavy artillery of the immune system.
Your immune system, really a marvel of biological engineering, has two main arms, broadly speaking. First, there are the humoral components, where specialized white blood cells called B lymphocytes, or B cells, synthesize and secrete circulating antibodies. These are the immunoglobulins we're about to explore.
The antibody producers.
Second, they're the cell-mediated components involving different white blood cells called T lymphocytes, or T cells. These T cells are crucial for various tasks, including defending against malignant cells, eliminating virus-infected cells, orchestrating hypersensitivity reactions, and mediating graft rejection.
Direct cell-to-cell combat and regulation.
Right. And to put it simply, a key difference is how they respond. B and T cells respond in an adaptive manner. This means they don't have pre-programmed responses for every single possible invader out there. Instead, they learn and create highly targeted, specific responses for each unique threat they encounter. It's like developing a custom-built weapon for every distinct challenge.
Memory and specificity are key features.
Exactly. The innate immune system, by contrast, is your body's rapid, more non-specific defense force. It has fixed components like phagocytic cells, macrophages, neutrophils, natural killer NK cells, and the complement system we'll touch on. These provide a generalized, immediate response that acts as a crucial first line of defense, holding the fort until the adaptive system gears up.
Innate is fast and broad. Adaptive is slower, but highly specific and powerful.
Good distinction.
Now, building on that, what's truly fascinating is the fundamental structure of these immunoglobulins, these antibodies. They are complex oligomeric glycoproteins, meaning they're made of multiple protein chains with sugars attached. They're composed of precisely linked heavy and light L polypeptide chains, originally designated by their migration rate during SDS polyacrylamide gel electrophoresis. The heavy ones move slower.
Heavy and light chains, got it. How many types are there?
Humans have 5 main classes or isotypes of immunoglobulins, each with distinct characteristics and functions: IgA, IgE, IgU, and IgM.
IgA, D, E, G, M.
No, IgG is by far the most abundant isotype in serum, typically consisting of 2 identical light chains, each weighing around 23 kilodaltons, and 2 identical heavy chains, which are larger, ranging from 53 to 75 kilodaltons, depending on the subclass. These 4 chains are linked together by a network of disulfide bonds, forming that classic Y-shaped configuration we often see depicted.
The characteristic Y shape.
Right. Other forms, like IgA and IgM, can form higher oligomers. IgA often exists as a dimer, 2 Y units joined together, or sometimes a trimer, particularly in secretions. IgM typically exists as a much larger pentamer, 5 Y units joined together in a circular fashion, giving it 10 antigen binding sites. This multivalency adds to their functional diversity, particularly for IgM's role in activating complement.
So dimers and pentamers too. And the classes differ in their heavy chains.
Exactly. Each class or isotype of immunoglobulin contains a different type of H chain, denoted by a Greek letter corresponding to the class name.
Alpha for IgA,
Yeah.
Old delta for IgD, epsilon for IgE, Kala for IgG, and mu for IgM. Okay, Ig heavy chains define the class.
Correct. The coated H chains of IgG, for example, are intricately organized. They have an amino-terminal variable region, VH, which is involved in antigen binding, and then 3 constant regions, CH1, CH2, CH3. IgM and IgE heavy chains, out in A, have an additional 4th constant domain, CH4, instead of a distinct hinge region.
Variable and constant regions. What about the light chains?
Light chains, meanwhile, come in 2 main types, kappa and lambda. These differ primarily in their constant regions, Cl. A single complete immunoglobulin molecule always contains either 2 identical key chains or 2 identical chains. You never find a mixture of kappa and lambda within the same antibody molecule. This ensures structural uniformity within any given antibody.
So 2 heavy, 2 light, either kappa or lambda light chains.
Precisely. Now, this raises an important question. What determines an antibody's specific job beyond just binding an antigen? Where do the functional differences between classes come from?
Yeah, why have different classes?
The constant regions of the heavy chains, particularly the CH2 and CH3 domains, and CH4 for IgG, are crucial here. These regions, often collectively called the FC region, fragment crystallizable, determine the class-specific effector functions. These functions include things like complement fixation, activating the complement cascade, binding to FC receptors on various immune cells like macrophages or NK cells, triggering phagocytosis or cell killing, and transplacental passage, allowing maternal IgG to cross the placenta and protect the fetus. The SEC region dictates how the antibody interacts with the rest of the immune system after it binds its target.
Ah, so the top of the Y binds the antigen, the bottom tail determines the action.
That's a great simplification, yes. The Fab fragments, fragment antigen binding, contain the variable regions at the tips, and the FC fragment forms the stem and dictates function.
Now, what's truly remarkable, you said, is the astonishing precision of that antigen binding at the tips of the Y.
Absolutely. Each typical IgG molecule contains 2 identical bivalent antigen binding sites, meaning it can bind 2 identical antigen molecules simultaneously. These binding sites are strategically located right at the tips of the Fab arms of the Y shape. Each site is formed by the interaction of the variable domain of a heavy chain, VH, and the variable domain of a light chain, VL. These domains fold together to create a unique surface that precisely binds to a specific molecular shape on an antigen called an epitope.
VH and VL together make the binding pocket.
Exactly. And the hinge region, which is found in IgG, IgA, and IgD, located between the CH1 and CH2 domains of the heavy chains, is particularly interesting. It's a flexible, somewhat exposed region that allows the 2 Fab arms to move relatively independently of each other, like wings.
Why is flexibility important there?
This flexibility facilitates optimal binding, especially when the antibody needs to bind to 2 separate antigen molecules or epitopes that might be spaced differently on a pathogen's surface or on different pathogens. It allows the antibody to adapt its reach, ensuring maximum binding efficiency. This flexibility is also why this region is particularly susceptible to cleavage by certain proteases like pepsin or papain, which historically allowed researchers to separate the Fab and FTC fragments for study.
Ah, Useful for research, too.
Yes, and when multiple antibodies recognizing different epitopes on the same pathogen are present, this flexibility allows for the formation of large antibody-antigen clusters or lattices. These large complexes are then readily recognized and efficiently engulfed and disposed of by phagocytic leukocytes, essentially tagging the invaders much more effectively for destruction.
Cross-linking makes a bigger target.
Precisely. Now, to further elaborate on this incredible binding specificity, within those variable regions of both the light and heavy chains, VL and VH, are even smaller regions called hypervariable regions. These are absolutely critical for determining the precise binding specificity.
Hypervariable, meaning they vary a lot.
Exactly. These are short sequences, typically only 5 to 10 amino acid residues long, that exhibit the highest degree of variability between different antibodies. Structurally, they project as loops from the surface of the immunoglobulins' variable domains, forming the actual contact surface for the antigen. These hypervariable regions are also often called complementarity determining regions, or CDRs, specifically CDR1, CDR2, CDR3, because their collective shape is complementary to the specific epitope they bind.
CDRs are the actual business end of binding.
They are the critical contact points.
Okay, here's where it gets really interesting and truly elegant, you said. The essence of these antigen-antibody interactions is profound mutual complementarity, like a lock and key.
Very much like a molecular lock and key system, or perhaps even better, like two perfectly molded surfaces fitting together. The surfaces created by the CDR loops precisely match the shape and chemical properties of the specific epitope on the antigen. Only the perfect fit will result in strong binding.
What holds them together?
This incredibly specific binding involves multiple, simultaneous, non-covalent interactions acting in concert. These include hydrogen bonds, ionic interactions, salt bridges, hydrophobic interactions, and van der Waals forces. Individually weak, but collectively, these interactions create a very strong and highly specific bond between the antibody and its target epitope.
Lots of little interactions add up to strong binding.
Exactly. This exceptional binding affinity, strength, and specificity, precision, of immunoglobulins for their target antigens comes from the remarkable ability of activated B cells to uniquely configure these hypervariable regions, these CDR loops of the immunoglobulin light and heavy chains, to perfectly complement a specific epitope encountered during an immune response. It's truly a marvel of biological engineering and adaptation through genetic rearrangement and mutation.
Okay, that leads directly to the big paradox. How can so few genes produce such immense diversity? You said the human genome has fewer than 150 actual immunoglobulin genes, yet each person can synthesize perhaps a million, maybe even billions, of different antibodies, each specific for a unique antigen.
Right. It's a biological conundrum that clearly defies the traditional one gene, one protein rule taught in basic biology. It reveals a far more dynamic and creative genetic system at play.
So how is it done?
Instead of having a separate gene for every antibody, immunoglobulin diversity is generated by sophisticated combinatorial mechanisms. These involve mixing and rearranging a finite pool of genetic information encoded in gene segments in a multitude of different ways during B-cell development.
Gene segments, like building blocks.
Exactly like building blocks.
Yeah.
Antibody diversity arises, in large part, from the way the coding sequences for each immunoglobulin chain are distributed among multiple different gene segments within the genome. For light chains, in the germline DNA before rearrangement, there are multiple different variable VL gene segments, several joining gene segments, and one or more constant CL gene segments.
VJC for light chains?
Okay. For heavy chains, it's similar but with an extra piece. Multiple variable VH gene segments, several diversity D gene segments, multiple joining gene segments, and then the constant CH gene segments for each isotype.
BDGC for heavy chains.
Oh, got it.
During B-cell development in the bone marrow, a process of DNA recombination occurs. For a light chain, one VL segment is randomly chosen and joined to one J segment. For a heavy chain, one D segment joins to one J segment first, and then one VH segment joins to that DJ unit. This creates a unique rearranged VJ light chain, or VDJ, heavy chain, exon.
So random combinations of V, D, and J segments.
Precisely. The human genome contains multiple functional versions of each of these V, D, and J segments. By randomly combining one V, one D for heavy chains, and one J, a vast number of unique variable region combinations can be generated from this limited set of germline segments. This combinatorial joining is a major source of antibody diversity before the B cell even encounters an antigen.
That creates a lot of possibilities already, is that?
Oh no, there's more. To truly unpack this diversity, let's explore another crucial mechanism that happens after a B cell is activated by an antigen. Somatic hypermutation, driven by a remarkable enzyme called activation-induced cytidine deaminase, or AID.
AID, what does it do?
AID works specifically in activated B cells, primarily in specialized structures called germinal centers in lymph nodes. It massively increases the frequency of point mutations, but only within the rearranged immunoglobulin V genes, the VJ and VDJ segments. It does this by catalyzing the chemical conversion of cytidine, C bases in the DNA, to uracil, a base normally found only in RNA.
C to U in DNA, that sounds like an error.
It is an error, and the cell's DNA repair machinery tries to fix it, but often introduces other mutations in the process. These are somatic mutations, meaning they occur in individual B cells during the immune response and are unique to the descendants of that particular B cell clone. They are not passed down through the germline.
So mutations happen deliberately in the antibody genes.
Yes. This process of somatic hypermutation creates unique B cell subpopulations, each with slightly different mutations in their V genes. This leads to the synthesis of immunoglobulins with subtly differing and often improved antigen specificities and affinities compared to the original antibody. B cells producing higher affinity antibodies are then preferentially selected to survive and proliferate, a process called affinity maturation.
Darwinian selection at the cellular level. Amazing.
It truly is.
But you mentioned earlier this process can go wrong. Autoimmunity.
Yes, that's the potential downside. To connect this to a broader context, in some pathological states, AID's powerful mutagenic action can accidentally lead to the generation of autoantibodies. If mutations unfortunately cause an antibody to recognize one of the body's own molecules, a self-antigen, it can trigger an autoimmune response. These are antibodies that tragically target the body's own endogenous components, leading to the phenomenon we discussed earlier called autoimmunity, where the immune system attacks itself.
The double-edged sword of mutation.
Indeed. And there's yet a third crucial mechanism for generating antibody diversity, happening during the VDJ recombination process itself. Junctional diversity.
Functional, at the joins.
Exactly. When the V, D, and J gene segments are cut and pasted together by the recombination machinery, involving enzymes RAG1 and RAG2, the process isn't perfectly precise at the joining points. Random numbers of nucleotides can be added by an enzyme called terminal deoxynucleotidal transferase, TDT, or deleted by exonucleases, right at the junctions between the segments.
So sloppy joining adds randomness.
It introduces significant randomness, particularly in the CDR3 region, which is formed by these junctions and is often the most critical region for antigen contact. Like AID-generated mutations, the changes introduced by junctional diversity are also somatic in nature, occurring only in the developing immune cells, and they massively amplify the potential antibody repertoire, creating even more unique binding sites.
Combinatorial diversity, junctional diversity, somatic hypermutation, incredible layers generating specificity.
Exactly. It's a multi-layered strategy to generate a vast B-cell repertoire capable of recognizing almost any potential foreign structure.
So what does this all mean when your immune system encounters a new threat and starts making antibodies? We often see different classes of antibodies appearing over time, right? Class or isotype switching.
Yes, that's another key feature of an adaptive humoral immune response. In most responses, antibodies of different classes, isotypes, are generated, but they all target the exact same specific epitope on the antigen. However, these different classes typically appear in a specific chronological order following exposure to the immunogen.
Which comes first?
For instance, the appearance of antibodies of the IgM class usually precedes that of the IgG class during a primary response.
IgM first, then IgG. How does the switch happen? Does the B cell start over?
No, it's more clever than that. Class switching involves combining the same rearranged VDJ, heavy chain, or VJ, light chain region, the part determining antigen specificity with different heavy chain constant region genes.
So the specificity stays the same, but the tail changes.
Precisely. Initially, a newly activated B cell typically expresses IgM and IgD on its surface. The rearranged VDJ segment is spliced to a constant region mRNA. Later, often under the influence of signals from T helper cells, the B cell can undergo a further DNA recombination event called class switch recombination, CSR. This process physically loops out and deletes the DNA encoding the C and C constant regions, bringing the rearranged VDJ segment adjacent to a different downstream constant region gene, like Co for IgG, Ca for IgA, or Co for IgE.
So it cuts out the old constant region DNA and joins the VDJ to a new one?
Correct. This allows the B cell to produce IgG, IgA, or IgE, but crucially, the VH region, and therefore the antigen specificity, the idiotype, remains identical to that of the original IgM molecule produced by that B cell lineage.
Wow. So it keeps the targeting system but swaps out the weapon system attached to it.
That's a perfect analogy. It allows the immune system to tailor the effector functions of the antibody response, mediated by the FDC region, to the specific type of pathogen or location of infection while maintaining the crucial antigen specificity. Combining the light chain with a good heavy chain makes Ig, good for complement, crossing placenta, while combining it with an heavy chain forms IgA, crucial for mucosal immunity at surfaces like the gut and lungs. Immunoglobulin molecules that possess identical hypervariable and variable domains, and therefore identical epitope specificity, regardless of their class, are said to share a common idiotype.
Idiotype is the specific binding site configuration.
Got it.
Now, what's particularly transformative here, moving from basic science to application, is the evolution of antibodies as incredibly powerful tools in medicine and research.
Monoclonal antibodies, right.
Well, let's start with the traditional approach. Traditionally, injecting an antigen into an animal, like a rabbit or goat, produced a heterogeneous mix of antibodies in the animal serum. These are referred to as polyclonal antibodies. Polyclonal meaning many different clones of B cells responded.
Exactly. These antibodies were directed against various different epitopes on that single antigen molecule. While useful for some applications, like certain diagnostic tests or historically for passive immunization, these weren't precisely targeted, and the resulting antiserum contained all sorts of antibodies produced by the host, not just those against the specific antigen of interest. Purity and specificity were limited.
Okay. A mixed bag.
Right. Here's where it gets truly revolutionary. The development of monoclonal antibodies in the 1970s by Kuhler and Milstein, which won them the Nobel Prize.
The hybridoma technology.
Precisely. Homogeneous monoclonal antibodies, ABs, target not just a single antigen, but a single epitope on its surface. This allows for unparalleled specificity and consistency, which revolutionized research, diagnostics, and especially therapeutics.
How are they made again? The fusion process?
Yes. They are generated by first isolating antibody-producing B cells from the spleen of an immunized mouse or other suitable animal. These B cells are then fused with immortal mouse myeloma cells, cancer cells derived from plasma cells that can grow indefinitely in culture. This fusion creates immortalized hybridoma cell lines.
Combining antibody production with immortality.
Exactly. Each hybridoma cell line originates from a single B cell clone and therefore produces and secretes only one specific type of antibody, a monoclonal antibody. These hybridoma lines are then meticulously screened using various assays to identify and isolate the specific clone that produces the desired antibody with the required specificity and affinity. That clone can then be grown in large quantities to produce vast amounts of the pure monoclonal antibody.
A factory for one specific antibody? Amazing. But mouse antibodies aren't ideal for treating humans, right? The immune system might react.
That's a critical point. For therapeutic applications, such as fighting coronaviruses, treating cancers, or managing autoimmune diseases, these original murine mouse antibodies often cause problems in patients. The human immune system recognized them as foreign, leading to the production of human anti-mouse antibodies, HAMA response, which could neutralize the therapeutic antibody or cause allergic reactions, sometimes severe.
Okay, so how do they fix that? Humanization.
Yes. Modern therapeutic antibodies are carefully humanized using sophisticated genetic engineering techniques. This typically involves precisely grafting the CDRs, the hypervariable loops that form the actual antigen binding site from the original murine antibody, onto the framework regions of a human immunoglobulin molecule, VH and VL. Sometimes the entire variable domain, VH and VL, is fused to human constant regions, creating a chimeric antibody, or more complex humanization strategies are used.
Replacing most of the mouse parts with human parts, but keeping the essential binding loops.
Exactly, and to highlight the critical importance of this humanization, it markedly reduces the immunogenicity of the antibody. It makes it look much more human to the patient's immune system, significantly diminishing the chances of HAMA responses, severe anaphylactic reactions, or other adverse immune reactions. It's a crucial step in making antibody therapy safer, far more effective, and widely applicable for treating human diseases, minimizing the risk of the patient's own immune system rejecting the therapeutic agent.
Making them stealthy therapeutic agents. Hashtag tag 9. The complement system, the innate immune support. Okay, Fantastic overview of antibodies. Finally, let's delve into the complement system. You mentioned it earlier as part of the innate immune system, but also interacting with antibodies. It serves as a vital innate immune support system, right?
Yes, think of the complement system as the backup,
Well,
the crucial support crew or amplifier for both innate and adaptive immunity.
amplifying the response. How is it innate?
It's considered innate because its core components are generally fixed, genetically determined proteins circulating in an inactive state. They provide a ready-made defense that doesn't need to learn to recognize new threats in the same way B and T cells do. But it's also incredibly versatile.
How so?
It can be activated not only directly by certain microbial surfaces, part of innate immunity, but also, very importantly, by adaptive immune components, particularly by antibody-antigen complexes. This is where the name comes from. It complements or enhances the activity of antibodies and other immune functions. It's a critical bridge between the two arms of your immunity.
Okay, innate but bridges to adaptive. How does it actually work? You mentioned cascades earlier, like blood clotting.
Exactly. That leads to an important question. How does it work, given its complexity? Like the blood clotting cascade, the complement system consists primarily of a set of circulating proteins, many of which are Zymogen's inactive proproteins or proenzymes. These are called complement factors, designated C1, C2, C3, etc., plus others like factor B, factor D.
Where are they made?
They are primarily synthesized by various cell types, with hepatocytes in the liver being a major source for many components. Macrophages, monocytes, and even intestinal endothelial cells also contribute. These factors circulate harmlessly in their dormant state until activated.
Waiting for a trigger.
Yes. Activation occurs via proteolytic cleavage, which then triggers a precise cascade of further proteolytic activation events. One activated factor cleaves and activates the next in a strictly ordered sequence. This amplification cascade ultimately produces one or more powerful protective end products designed to eliminate pathogens. The most famous of these is the formidable membrane attack complex, or MAC.
The MAC attack. Okay, how is it triggered? Let's start with the classical pathway you mentioned, involving antibodies.
Right. The classical pathway is typically triggered when an antibody, specifically IgM or certain subclasses of IgG, binds to an antigen, for example, on the surface of a bacterium. This antibody-antigen complex then binds to and activates the first component of complement, C1.
C1 binds the antibody tail.
Essentially, yes. C1 is actually a complex itself. C1q, C1r, C1s. Binding of C1q to the Fc region of the bound antibody activates the protease activity of C1r and C1s. Activated C1s then precisely cleaves two other complement factors, C4 into C4a, a small inflammatory mediator, and C4b, a larger fragment that binds covalently to the target surface near the antibody, and C2 into C2a, which joins C4b and C2b, another small fragment.
The C4b and C2a stick together.
Yes. The C4b and C2a fragments associate together on the target surface to form a new, crucial enzyme complex called the classical pathway C3 convertase, C4b2a.
The 3 convertase. Its job is to cut C3.
Exactly. This C3 convertase is a pivotal enzyme. It cleaves numerous molecules of complement MHC3, the most abundant complement protein is C3a, another small inflammatory mediator, and anaphylatoxin, and C3b, a larger fragment. C3b is the major opsonin of the complement system.
Opsonin, meaning it tags the pathogen.
Yes, it coats the pathogen surface, making it much easier for phagocytic cells like macrophages, which have C3b receptors, to recognize and engulf the invader. Opsonization.
Okay, tagging for eating. What else happens?
Some of the C3b generated then binds to the existing C3 convertase, C4b2a complex. This forms a new enzymatic complex, the C5 convertase, C4b223b. Its job is to cleave the next component, C5.
Cutting C5 now.
Right. The C5 convertase cleaves C5 into C5a, a very potent inflammatory mediator and chemoattractant for neutrophils, and C5b, the larger fragment. C5b is the initiating component for the final step, assembly of the membrane attack complex.
The MAC assembly starts with C5b.
Correct. C5b, while briefly membrane-associated, sequentially binds complement factors C6, C7, C8, and finally multiple copies, up to 18 of C9.
C5b 6, 7, 8, 9.
Yes. This entire assembly, C5b9, forms the membrane attack complex. C7, C8, and particularly C9 insert themselves into the lipid bilayer of the pathogen's plasma membrane, forming a transmembrane channel or pore.
Punching a hole in the bug.
Exactly. The MAC kills susceptible bacteria, especially gram-negative bacteria with outer membranes, by opening this pore in their membrane. This disrupts the osmotic balance, allowing water and ions to rush in, causing the cell to swell and lyse burst open. The remains are then cleaned up by phagocytes. Meanwhile, those small fragments generated earlier, C3a and C5a, serve as potent chemoattractants, recruiting more leukocytes like neutrophils and macrophages to the site of infection and powerfully stimulating a localized inflammatory response, further aiding in pathogen clearance. They're alarm signals.
So direct killing via MA, tagging via C3b, and alarm signals via C3a, C5a. Powerful system.
Very powerful. And what's particularly fascinating here, providing another layer of specificity, is a precise molecular detail involving those reactive thioester bonds we mentioned earlier in C3 and C4.
Ah, the thioesters again. What do they do here?
Similar to immunoglobulins, C3 and C4 contain a hidden internal thioester bond. When C3 and C4 are cleaved by their respective convertases, C1's for C4, C3 convertase for C3, this highly reactive thioester bond in the C4b and C3b fragments becomes briefly exposed.
A hidden reactive group pops out.
Yes. It can then rapidly react with hydroxyl or NH2 groups present on molecules on the nearby cell surface, such as bacterial surface polysaccharides or proteins. This reaction covalently attaches the C4b and C3b fragments directly to the pathogen surface.
Why is that covalent attachment important?
It ensures that the subsequent steps of the cascade, including the formation of the C3 and C5 convertases and ultimately the UNAC assembly, occur directly on the pathogen surface where they are needed, rather than diffusing away into the fluid phase. It covalently anchors the complement attack machinery right onto the target, precisely facilitating MSC assembly nearby and maximizing its efficiency against the invader while minimizing collateral damage to nearby host cells. It's molecular targeting.
Very clever. Now, are there other ways to start this cascade besides antibodies? You mentioned the lectin pathway.
Yes. The complement system can also be activated via the lectin pathway. Okay, let's unpack this. This pathway is like a sugar-seeking missile, recognizing specific carbohydrate patterns often found on microbial surfaces, but not typically on host cells.
Sugar patterns as triggers.
Okay.
Certain bacterial polysaccharides, particularly those rich in mannose residues, bind to a specific circulating protein complex called mannose-binding lectin, or MBL, sometimes also called mannan-binding protein, MBP.
MBL binds microbial sugars. Then what?
MBL is structurally related to C1q and circulates in complex with MBL-associated serine proteases MASP1 and MASP2, which are analogous to C1r and C1s. When MBL binds to the microbial surface sugars, the MASPs become activated. Activated MASP2 then cleaves C4 and C2, just like C1s does in the classical pathway.
So MBL and ASP essentially replaces C1.
Exactly. It generates the same C3 convertase, C4b2a. From that point onwards, the rest of the cascade mirrors the classical pathway exactly. Cleavage of C3, formation of C5 convertase, and assembly of the MAC.
So it's an antibody-independent way to activate the same downstream pathway. Correct. A lectin, by the way, refers broadly to any protein that binds specifically to carbohydrates, polysaccharides. Most lectins are highly selective for particular sugar structures. MBL is specifically designed to recognize terminal mannose and fucose residues, which are commonly found in the mannans on the surface of many gram-positive and gram-negative bacteria, some viruses, fungi, and protozoa, but are less common on mammalian cell surfaces.
Recognizing foreign sugar patterns.
Yes. Structurally, MBL circulates as large multivalent complexes, typically ranging from 400 to 700 kilodaltons. They consist of 4 or more copies of a homotromeric core unit, 3 identical polypeptide chains. Each subunit has a collagen-like domain that intertwines with others, and a carbohydrate-binding domain located in a globular head that projects outward. This forms a branch structure, somewhat resembling an immunoglobulin or C1q, allowing it to bind efficiently to multiple repeating sugar targets on a pathogen's surface, triggering the cascade.
Okay, classical and lectin pathways. The alternative pathway.
Yes, there is a third major activation route, the alternative pathway. This pathway is particularly important for immediate innate defense, as it doesn't rely on antibodies or MBL binding first.
How does it start then?
It's initiated by the spontaneous low-level hydrolysis of the internal thioester bond within C3 molecules circulating in the plasma. This process is sometimes referred to as C3 tickover.
So C3 just randomly activates itself slowly.
At a very low rate, yes. This generates a small amount of C3b, or a C3b-like form called C3H2O. If this spontaneously generated C3b happens to land on a host cell surface, it's usually rapidly inactivated by regulatory proteins present on host cells, like factor H and factor I. However, if it lands on a microbial surface, which typically lacks these regulatory proteins,
it can stick around.
Yes. On a non-self or activating surface, the C3b can bind another complement factor called factor B. The resulting C3bb complex is then cleaved by a circulating protease called factor D. This cleavage releases a small fragment and leaves the larger blab fragment attached to C3b, forming the C3bb complex.
C3bb is in another convertase.
Exactly. The C3BBB complex is the C3 convertase of the alternative pathway. It's functionally equivalent to the C4B2A complex of the classic lectin pathways. Its job is to cleave more C3 into C3A and C3B.
Ah, so it creates more C3b, which can then form more C3bbB, an amplification loop.
Precisely. This forms a powerful positive feedback amplification loop right on the microbial surface, rapidly coating the pathogen with C3b for opsonization and generating more C3 convertase activity. The alternative pathway C3 convertase, C3BBB, is inherently unstable, but it can be stabilized by another protein called propertin, factor P. Eventually, when enough C3b is deposited, some of it can bind to the C3BB complex itself, forming C3BBB3B, which acts as the C5 convertase of the alternative pathway.
And the C5 convertase cuts C5, leading to the anaemia.
Correct. So the alternative pathway provides a rapid antibody-independent mechanism for complement activation and amplification directly on microbial surfaces, serving as a crucial first line of innate humoral defense. #10. Dysfunctions of the immune system, pathologic consequences.
Okay, we've covered antibodies and complement, these amazing defense systems, but let's end by bringing it back to the critical impact when these systems go wrong. The dysfunctions of the immune system, which can lead to various pathologic conditions affecting millions of people. First, let's consider immunodeficiency. This is essentially when the immune system is too weak, right?
Yeah.
Caused by deficits in producing vital immunoglobulins or perhaps complement factors.
Exactly. It leaves the body vulnerable. That leads to an important question. Why does a weak immune system lead to such profound vulnerability?
Yeah. What are the consequences?
It leaves these immunocompromised individuals highly susceptible to a wide range of bacterial, fungal, or viral infections, sometimes with pathogens that wouldn't trouble a healthy immune system. Their body simply lacks the necessary tools, the antibodies, the complement activity, the specialized T cells to mount an effective defense.
So they get frequent or severe infections. What causes this deficiency?
Many factors can contribute to this depression in immune effectiveness. There can be primary immunodeficiencies caused by genetic abnormalities, such as X-linked agammaglobulinemia, where B-cell development is blocked, markedly affecting IgG production. There are also genetic deficiencies in various complement components. Then there are secondary immunodeficiencies which are acquired. These can be caused by exposure to certain toxins, viral infections like HIV, which specifically destroys helper T cells, severe malnutrition, proteins are needed to make immune components, neoplastic transformation, cancers like leukemia or lymphoma affecting immune cells, or, very commonly today, the medical use of immunosuppressant drugs.
Why use immunosuppressants?
They are essential for preventing rejection after organ transplantation or for managing severe autoimmune disorders by intentionally dampening the overactive immune system. But a major side effect is increased susceptibility to infection.
Unnecessary trade-off sometimes.
Often, yes.
Okay, so that's immunodeficiency, the system being too weak. So what's the core understanding here for your health on the flip side? Autoimmune disorders. This is when the system is tragically misdirected.
Precisely. This is when your body's incredibly sophisticated immune system, designed to protect you, tragically turns on itself. It fails in its crucial task of immunological tolerance, the ability to differentiate between the body's own self components and foreign non-self invaders.
That non-self recognition breaks down.
Yes. This profound recognition error triggers an autoimmune response, where lymphocytes and antibodies start attacking the body's own healthy tissues and organs as if they were foreign threats.
What kind of damage can result?
The resulting damage can be cumulative and progressive, leading to chronic conditions like rheumatoid arthritis, which relentlessly attacks the joints, or multiple sclerosis, where the immune system attacks the myelin sheath, insulating nerve fibers in the brain and spinal cord. Alternatively, the damage can be acute and devastatingly rapid, resulting in the complete destruction of specific cells, as tragically seen with the insulin-producing pancreatic beta cells in type 1 diabetes.
Different patterns of attack. How common are these disorders?
Autoimmune disorders are surprisingly prevalent, collectively affecting potentially 3 to 5% of the population in Western countries, maybe around 3 in every 100 persons in North America, depending on how you count them. They represent a major health burden, highlighting the fragile balance of self-tolerance required for a healthy immune system.
Can you list some common examples again?
Sure. The list is long and diverse, including conditions like Graves' disease, causing hyperthyroidism, rheumatoid arthritis, Hashimoto's thyroiditis, causing hypothyroidism, vitiligo, loss of skin pigment, type 1 diabetes, pernicious anemia, attacking stomach cells needed for B12 absorption, multiple sclerosis, systemic lupus erythematosus, SLE, affecting multiple organs, Sjogren's syndrome, attacking salivary and pituitary glands, myasthenia gravis, attacking nerve muscle communication, Addison's disease, attacking adrenal glands, and scleroderma, causing hardening of skin and connective tissues. It's a wide spectrum of diseases driven by immune self-attack.
A huge range of conditions, all stemming from that fundamental failure to recognize self.
That's the underlying principle, yes, though the specific triggers and mechanisms vary greatly between diseases.
Okay, we've really taken a comprehensive deep dive today, haven't we, into this incredible, intricate world of plasma proteins and immunoglobulins.
We certainly have covered a lot of ground.
Yeah, we started right from their fundamental roles in maintaining fluid balance and transporting vital nutrients, then moved through their complex involvement in inflammation, that precise dance of iron homeostasis, and finally, the sophisticated adaptive machinery of the immune system, antibodies, and complement. You've now really seen how these microscopic players are absolutely essential for your health.
And conversely, how their dysfunctions, when they go awry, can lead to serious and often debilitating diseases.
It's truly a testament to the biological elegance and the complexity operating silently within us all the time.
Absolutely. What's truly fascinating here, I think, is the sheer elegance, the incredible precision, and the profound complexity of these biological systems. Understanding them, even at this level, reveals just how intricately your body works moment by moment to defend itself and maintain its delicate internal balance. It powerfully emphasizes that even seemingly simple elements of blood are, in fact, part of these highly regulated, interconnected networks where even a small change, a single mutation or deficiency, can have profound implications for your overall health. You now possess the foundational knowledge, really the depth of understanding of an elite professional in this area, giving you a truly deep appreciation of these vital components.
So what does this all mean for you, the listener, moving forward, armed with this much deeper level of understanding, knowing how precisely these systems operate? Maybe consider this. What emerging technologies or personalized medicine approaches could truly leverage our growing understanding of plasma protein dynamics, iron regulation, or immune system intricacies to perhaps better predict, prevent, or even reverse some of these complex diseases that plague so many?
The future possibilities are exciting.
They are. And maybe on a more personal note, how might your own daily lifestyle choices, things like nutrition, managing stress, physical activity, how might those subtly influence these silent, powerful networks operating within you? Could you be supporting their health and function or inadvertently hindering them? Something to think about.